around, not ahead. A peak is just a way to walk at the sun. The forces on a hill manifested by what you perceive as wind. Look around, not ahead. Look around, not ahead. The peak is just a way to walk at the sun. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill manifested by what you perceive as wind. Look around, not ahead. Look around, not ahead. The peak is just a way to walk at the sun. Have nowhere to look. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead.
Welcome back to the second day of Translate here in Berlin. We're very happy that you join us for the second day. I would like to start briefly with some technical details before leaving the floor to Dave Granger, who will warm you up with the content and topics. The first point that I'd like to address is that if you haven't registered yet, please register, that you have full access to all details of the meeting and the presentations. And we'll also put in this later the flow from the yesterday, as well as the recording of today's symposium. Second point is I would definitely like to thank the German Research Foundation and Science Translation, as well as Charité BAH, to make that possible. What I haven't really properly enough pointed out is going to be a core element of today. We are partnering here with the German Research Foundation, and in the German Research Foundation there is the Senats Commission, the SGKF, who is in charge of all research and political topics related to medicine, science and medicine in Germany. And for that commission, Jörg Schulz is going to moderate one of the sessions later. We also have the vice president of the German Research Foundation with us, Britta Siegmund, and from the head office, Eckhard Pichte, supporting that event. The second point that I wanted to mention, there is a chance to give your comments and suggestions using the Slido feature. If you scroll down, you can ask your questions. That was used yesterday. It could be also more intensively used. We have it here, the flow, and we will try to bring the questions to the podium and bring them here into the discussions. With that, I'd like to stop and hand over to Dave Granger, who is a good friend, sits in a wonderful snowy environment in the middle of the US, and between different skiing activities, eventually is very actively engaged in the topic of translation in life sciences. And Dave, how is it in Utah? And welcome here.
Thanks, Georg. What a great day here. New snow on the ground, and yeah, it's cold, but I'll try to warm everybody up. We had a great day yesterday, and just to remind everybody, I think that we can share some slides here with our roundup feature. And pull up some slides from the summary. So we had a number of so let me just simply say in the summary that we'd like to thank our sponsors. Next slide. And the sponsors, of course, make this whole event happen. And I want to feature them prominently and thank them very much for allowing us to have this global event. It's been a difficult time to get this all arranged, of course, in the given circumstances. And I'm so happy that our sponsors have been patient with our formatting and with the virtual delivery of the meeting. All right, next slide. With that, I'd like to go to the roundup, try to warm you up for another excellent day. In our first day yesterday, let me simply remind you that we had some take-home messages I'd like to briefly go through. Philanthropy was interesting as a new mode to promote freedom for academic translational efforts in a way that perhaps our industry or our federal funding mechanisms cannot. I just reminded you of the Janis Joplin lyrics from the Bobby McGee song, "Freedom is just another word for nothing left to lose." So keep that in mind when you think about freedom and philanthropy of freedom. Our second point, of course, is innovation ecosystem. And many speakers yesterday emphasized that as a correct and a very essential feature, that you have to inventory all of the proper components for your ecosystem, make sure that they are there, because just like a well-oiled Mercedes-Benz, the machine must comprise all of the correct pieces and operating in the correct functions. Team was also emphasized yesterday as a way to get your expert players on the team, because no innovation concept in translation can in fact move from the bench to the bedside or even back in these repeated iterative cycles without the teaming that's necessary to get all of the expertise in place. Then there was a very important point, and we'll hear this again today, about market pull for your emerging ideas versus technology push from the inventors. And that oftentimes the push is not seen as the appropriate maneuver, that you respond to market pull for your innovative incentives. Digital health was emphasized yesterday, and it will be also emphasized again today. And the issues of governance and data governance versus data privacy, patients' rights versus user privileges, and producing the necessary trust from patients in moving their data through management and exploitation. I think that we have to be vigilant of that. Another aspect of that was the digital medicine aspects of understanding patient harm from digital curation of their data, and producing the proper culture, the legal and the ethical and regulatory challenges associated with curating patient data in this environment. In terms of metrics for measuring translational success, there were many proposed yesterday. I think this is a very cloudy area, certainly one that this meeting is seeking to address. And one of the take-home messages yesterday was that, yes, there are many metrics for measuring success, but they're applied appropriately in different places of the process. They're not all just applied uniformly, but in fact, there are different metrics of success that must be levied at each stage of translational development. And then translational medicine should include a very close concerted joint training, joint mentoring, joint research and translation of PhDs with an MD culture. That is, medical doctors mixing with PhDs at all steps of the process so that they can appreciate their contributions and the relative value of the teaming effort there. In terms of discussing our consensus papers, reminders that there are 2 consensus papers that we're interested in developing with you as an audience. And there are many issues in those papers and diverse opinions and perspectives, and we welcome your input to those consensus papers, and you can find that link online, and there will be discussion at the end of today about those 2 papers. And then finally, the digital labs discussion from our colleague at the BIH in discussing the translational engine for German hospital networks and their digital startups that have come from that network, and perhaps the ability to extend that model beyond Germany to Europe and beyond Europe to the world. And finally, in closing yesterday, we had the challenge at Translate Issues looking at those 3 startup pitches from 3 different teams. And of course, I think the commonality for all of them was the ability to produce teaming, the ability to produce valuable IP and patents, and the regulatory challenges associated with the current digital age of spinning medical technology out. So with that then, we'll move to our first speaker of the morning, which is my good friend and colleague, Bruce Tromberg. Bruce has a very decorated career. He started in 1990 as a professor at the University of California, Irvine. He's a well-known laser spectroscopist. In fact, he was at one time one of the top 10 researchers funded by the National Institutes of Health in my country in the field of optical coherence tomography, OCT, for medical imaging. He became a professor in the Department of Biomedical Engineering and Surgery in 2002, and then became the director of the famous Beckman Laser Institute and Medical Clinic as of October 2003. Most notably, Bruce was selected to be the director of the National Institute of Biomedical Imaging and Bioengineering. We call it NIBIB, within our National Institutes of Health on January 7th of 2019. His appointment is at the level of the now famous Anthony Fauci, who is his colleague at the director of the National Institute of Allergy and Infectious Disease, and Bruce represents the equivalent position in a different institute. And they are both one of 27 possible NIH institutes and centers that comprise the most powerful federal funding body for medical research, I think, in the world. I think Bruce will talk about what I've asked him to talk about, which is the RADx initiative. And this initiative was formed on April 29th, 2020, this year, in response to the COVID-19 problem. And its entire mission is to speed the development, innovation, and commercialization of technologies for COVID-19 testing. And notably, this initiative has produced the first COVID-19 home test from the NIBIB's effort that has been FDA approved under the emergency use authorization approval on just December 15th of 2020. And there are 4 programs involved in RADx with many partners in a public-private partnership that has been unprecedented in accelerating the translation of technology addressing the current COVID crisis in our country, and I hope to spill and produce a global impact. So without further ado, Bruce is going to produce his talk from his computer, and I'd ask the organizers to help me share that screen with him to proceed. Hi, Bruce. Good morning.
Hi, Dave. How are you? Good to see you. And can everyone see my slide?
I can see you, and I can't see the slide yet, but they'll arrange that. You'll need to share. Can you share?
I am. It's up and sharing. I think hopefully
now we can.
They'll.
Great.
Great. Well, thank you for that very kind introduction. I'm going to remember that you mentioned me in the same breath with Tony Fauci. So Tony is pretty spectacular. I carry my Tony Fauci button around with me. I'm not wearing it at this moment, but I do have to show it off. And I have some Tony Fauci socks and a few other cool Tony Fauci things. It's been quite a ride here at the NIH, led by Tony and Francis Collins. And fortunately, we've been able to rely on their leadership and vision and experience. And so what I'll talk to you about today is what the Engineering Institute at the NIH. We're a relatively small institute, but we have lots of really important technologies we think that we can bring to bear on these problems. So I'm speaking to you from Bethesda. Greetings from my colleagues at NIDIB. And this picture's about a year and a half old. It reminds us of the time when we could gather together, and inspires us to think about the time in not too distant future when we can do this again. As Dave mentioned, he asked me to talk about all the unexpected features of the pandemic, the opportunity, the circumstances, and the consequences. So I'll try to weave all that together and tell this story about the RADx program. About a year ago, it was actually at the Photonics West meeting in San Francisco in early February. Many of us were tracking COVID around the world, and there was a travel ban that was just put in place from China, from Asia to the US. And we were really contemplating what could the bioengineering community do as we watched this growing infection around the world, and pondered whether it would eventually become a pandemic. And from a bioengineering point of view, we immediately took a look at, and Dave, you mentioned essentially ecosystems. And of course, the bioengineering communities around the world have invested in innovation and entrepreneurship in many different areas. For us, our biggest investments and strongest areas have been in imaging and artificial intelligence, digital health platforms, and diagnostic test technologies. So we began contemplating a strategy where we could potentially expand these approaches to address this evolving pandemic. And that was really then combined with quite an unexpected opportunity, which occurred in April of 2020. On April 24th, Congress, in their fourth appropriation, designated $1.5 billion to go to the NIH, and $500 million went to the NIDIB. That actually amount was more than our annual budget, and it was clear that this was a very, very unique and unexpected opportunity for us to have an impact. So we partnered with the Office of the Director, led by Francis Collins and colleagues, to create a program that we call Rapid Acceleration of Diagnostics. And as Dave mentioned, there are different versions of RADx. There's a tech component and a component that we call ATP, which is scaling up existing platforms. And between the two, we've had over $700 million to invest. There are also programs to direct technologies and testing technologies to underserved populations. That's RADx-UP, as well as very forward-looking approaches, RADx-RAD, which are several, still a few months or years away from coming to fruition, but important, potentially very innovative approaches. We immediately began forming partnerships across the government with notably BARDA, which is one of the US government agencies for advanced research technologies and projects that are specifically addressing issues of pandemics and infectious disease and outbreaks and disasters. And they invested another $300 million in a partnership with us at the NIH. And most recently, Congress appropriated $100 billion in December in a fifth supplement. So we had working capital and we had some existing structures, which I'll go into. We formed partnerships across the government with FDA, the Department of Defense, the Center for Medicaid Services, and the CDC, and began a regular cadence of meetings and setting goals. And those led to perhaps unexpected, but maybe in the end, not so surprising results, and I'll get into those just a second. We were able to launch this program just 5 days after that appropriation because we had been thinking about what we could do and had been preparing a structure in the event that some additional funding could come. If we look at the original goals at the outset, they were to expand testing technologies, the number of tests, their type, their accessibility, and optimize their performance, both technologic and operational performance, so that each of the tests could match better community needs. if you go back to the end of April or beginning of April, when we began to conceive of the program, the United States was doing about 250,000 tests per day. These were predominantly in laboratory settings. In fact, they were all in laboratory settings. And we envisioned the need to expand substantially point of care technologies and home-based technologies. And as you can see in the graph on the right, which is taken from the COVID tracking project, the tests per day in the United States have grown quite remarkably since April. This is predominantly reflecting laboratory tests, and we are now at about 2 million tests per day. But there are another several million tests per day that are coming from rapid antigen tests, most notably lateral flow assays, LFA, that are unreported. These tests are not connected into our digital health backbone or our systems for reporting with public health, and they're reflecting the change in abundance of testing technologies from laboratory-dominated, which we were in the early days and has been more or less the standard of care, to point-of-care testing, and now we'll see a dramatic expansion in home testing technologies. So in total, the United States is doing many more tests than what is reported on these websites that are tracking the actual tests that are performed. We were able to launch the program by building on an established network that we'd supported for 13 years. This is the Point of Care Technologies Research Network, or POCTERN. And just to highlight some of the features in this network, first of all, there are multiple universities that are working together and collaborating in order to develop and validate point-of-care technologies. The Central Coordinating Center is at CIMMT, Mass General Hospital. It includes Johns Hopkins, Georgia Tech and Emory, Northwestern, and UMass. We built into the network some additional features. First, a core to be able to validate every technology that would be evaluated and have an independent confirmation of the performance. We've had more than 50 projects come through the core, and several thousand human subjects participants are actually evaluated using these various technologies. We have a clinical studies core for standard trial design and assessing digital health platforms, single IRB, so we can rapidly do clinical studies, and a deployment core to address issues such as supply chain and manufacturing and develop end-to-end solutions so that the tests that are produced can actually get sold and distributed and implemented in settings that need them. So this network, with the addition of some nonprofits and more than 1,000 experts all over the world, have allowed us to review and fund concepts, test and validate every one of the technologies, and provide expert guidance for technologies that are making it through. The major engine or the vehicle for making all this happen is a classic innovation funnel, which we've reduced to practice on quite a remarkable scale. We had more than approximately 3,000 applications from all around the world that were started. We ended up having over 700 that were completed, and each application is reviewed and assessed for funding at different stages. So the initial review is a week-long deep dive. Some people have popularized this concept as a Shark Tank. It seems to have hit a resonant tone in the country as something that captures people's imagination. But it's really what I think many of you are very familiar with. We team every project up with another group of experts, about a half a dozen, who can help understand and guide each project and really determine what the project will need in order to be successful. That then is presented to additional review panels, and the company or the project is eligible for phase one funding up to about $1 million, which can last anywhere from 4 to 6 weeks. And in that period of time, each technology is de-risked, assessed in the validation core, and if those milestones are met, there's continued a next level of evaluation for phase two, where potentially companies can receive contracts that would be tens of millions of dollars that would help them in everything from regulatory to scaling up in manufacturing. And we've had 25 companies now, a little less than 4% of the total completed applications make it through to phase two. We've invested about $500 million, and I'll show you what the impact of all of that is in just a second. But the idea is if you start with a project and make it all the way into phase 2, you will receive regulatory approval through our close collaboration and interactions with the FDA and be ready for scaling up and distribution of the technologies. So these are some examples. There are 25 different companies that have received support of different sizes, from small businesses all the way through large companies. We have quite a strong representation of point of care and home-based approaches, both RT-PCR as well as lateral flow assays, quick antigen tests. Most notably, as Dave mentioned, one of these technologies made by Illum, which is partnered with QIAGEN, has an antigen lateral flow assay that's now been approved for over-the-counter use, and that's currently in wide-scale rollout across the country. There are laboratory tests that we're supporting as well. Some of them are entirely new approaches, like mobile labs, such as developed by this small startup from Wisconsin, Flambeau Diagnostics. And some of them are well-known, such as Fluidigm, which makes integrated fluidic chips primarily for laboratory use, but these have been transformed for their applications for COVID with an EUA in saliva testing. That single chip can do 192 assays in parallel, and their Biomark platform can do many of these chips in a day. You probably recognize other performers or other companies in this group as well. Some of them, for example, like the Broad Institute, have scaled up to be able to do 100,000 tests in a day, and they're continuing to scale and expand. In the upper left-hand corner, you can see some really interesting novel approaches, handheld RT-PCR devices with excellent performance that I'll talk a little bit more and compare that to lateral flow assays, but just as a way of comparison, you can see this is the handheld device from Mesa, and this is their cartridge that's used for every sample that's removable and disposable, and is really quite competitive just in terms of the price and the performance of those devices. This is the Illum assay, and it's got an integrated optoelectronics components in it that are built in application ASICs or application-specific integrated circuits. It's actually, the cost of goods are quite inexpensive, and it relies on quantum dots for readout and reporting. So lots of really interesting things happening here. Then there are also some very far forward-looking technologies. Not all of the technologies that we've supported can go to manufacturing and scale up under these types of time pressures. And we've tried to continue to support some of these through existing NIH networks. We have a seed network, which is providing additional project funding, industry coaching and mentoring and training resources with about 50 more of these projects. So this is the kind of unexpected impact and result that we've seen 5 months after launch. And normally, if you're familiar with the time course of translation, it takes anywhere between 5 and 10 years for companies to go from concept stages to wide-scale dissemination and manufacturing. We had already been able to contribute about 20 million tests in the month of September, and this has continued to grow so that in 2020, we had approximately a little over 90 million tests that were produced, and of these, 48 million of these have been sold.
Bruce, I think you're back. At least I can see you.
Okay.
Might have to share screen again. I don't know.
Okay.
There we go. Back on.
All right. Good. So let me just try to pick up. I'm not sure where I was lost, but we've produced about a little over 90 million tests in 2020. About 48 million of these have been sold. So our testing capacity always exceeds those are sold for a variety of reasons. By the end of the year, we were producing, or RADx supported companies were producing about 1 million tests per day, and roughly half of those have been sold and deployed. a number of emergency use authorizations. Overall, we've been able to support about 150 companies, 25 of these in phase 2. As we look forward into the coming year, we expect that there will be millions of over-the-counter lateral flow assays that are produced per day that will begin to appear. And by March, we're expecting around 2.5 million tests per day for RADx-supported efforts. We've had, as I mentioned, cores that were built into the program, and our deployment core is really tasked with trying to reduce that gap between tests that are produced and tests that are actually sold. And one of the key engines, a major operational component of this is this website.
So looks like we've lost Bruce again.
That are actually sold.
I can see him.
It's one of the key engines, a major operation. Am I back?
Yes, you're back.
Okay. Well, let me try to accelerate through the rest of this before we completely crash. I encourage everyone to take a look at the whenTotest.org website, which is developed by our deployment core at CIMMYT MGH. It helps you understand the needs of your organization and can help create a better view of what needs to be deployed in terms of mask wearing, the size of unmasked group activities, and the types of testing technologies and their frequency, as well as their cost, in order to mitigate and interrupt the spread of COVID in your organization. They are responsible for creating playbooks for different settings, connecting purchasers and vendors, coordinating our supply chain solutions, as well as collaboration with multiple groups outside of the creation of the technologies. And the validation core, as I mentioned, is evaluating every one of the technologies individually, very focused on new technologies, for example, to bridge the gap between the performance of conventional RT-PCR, handheld RT-PCRs, and handheld and point-of-care lateral flow assays. So there are a variety of different form factors for those lateral flow assays, but in general, their limits of detection are anywhere between 2 and 3, maybe even 4 orders of magnitude higher than those of PCR. So the new technologies include CRISPR, microfluidics approaches, a variety of different nanoparticles and single molecule strategies, ASICs and waveguides, but also this core is exploring how we can develop new protocols around technologies, for example, pooling point-of-care PCR, so that the cost-effectiveness of the point-of-care instrument actually is really quite high, and the roughly one can imagine having about $1 per person for effectively of a cost of these tests. If you can pool 10 or 15 people, a social pod, such as a classroom or a home, with a single cartridge. The clinical studies core is evaluating all of our platforms in clinical studies to look at real-world guidance on their use and how we can integrate digital health. So a quick snapshot of what some of these studies are guidance on the use of sequential lateral flow assays and assessing whether or not multiple lateral flow assays can give you equivalent understanding and performance to a single RT-PCR. We're also looking at digital health platforms and currently planning a large, potentially quite large public health study that would be targeted towards one or more cities where inexpensive at-home assays would be distributed, and we could then assess whether or not regular frequent tests can break the chain of transmission of the virus. To help facilitate all of this, we've also been working at the same time on digital health platforms that are companion pieces with the tests. They can provide instruction on how an individual would perform a test and interpret it, track symptoms and history, and be connected, provide a digital record of the test result, also connect to electronic health records, provide the ability to do contact tracing and proof of health status, and send information out to public health organizations. So let me try to summarize. We could probably replace that D with a T in a sense, RADx, although it doesn't sound quite as good, but rapid acceleration of translation. This is something that the program has been able to do in a matter of months. It takes many, many years. And really broadly speaking, there are two types of translations. One is bench to bedside. So we've taken research lab technology, as I mentioned, nanoparticles, microfluidics, waveguides, and ACE from what many of us are used to at the lab bench and have encouraged it to move out into clinical settings, laboratory point of care and at-home use. And the second type of translation that's occurring is concept to market. So ideas to prototypes, prototypes to scale up, and boutique kinds of scale ups. For example, I showed the Mesa Acula technology. They've gone from just a few thousand tests per day to 20,000 tests per day, and will continue to expand on that number. And we've had wonderful representation from the innovation and entrepreneurial community. Small businesses are by far the largest applicants to RADx. And between small businesses and mid-sized businesses, we have really the bulk of the activities that are being supported. And we've been able to do this by leveraging and expanding an existing network. So we've been able to new processes in place to allow us to provide resources, funds to every one of these companies at really unprecedented speed. And through that innovation funnel time pressure, get really unprecedented impact. There's an urgent need for purpose-driven technology. I think we've really underscored that. Off-the-shelf solutions are not always ready when you encounter unexpected circumstances such as a pandemic like this. We've really brought together, I think, bioengineering and the technology community with new and perhaps non-traditional partners in public health and policymaking, which I think underscores a need to do this more in the future. And we have ongoing challenges, of course. We've made over $1 billion investment in diagnostic technology, and we need to make sure that this is transferred and continues to move so that it's impacting other diseases and future pathogens. And finally, for the United States in particular, our regulatory, healthcare, and reimbursement systems are not really optimized for the idea of screening and surveillance. They're really designed for detecting disease in individuals. So this is perhaps an inflection point, and we'll see how economically sustainable this is now that we've helped stimulate this type of process. So I'll stop there, and apologies for all of the interruptions, and maybe we have time for a few questions.
Well, thanks, Bruce. That's awesome. We do have 5 minutes, and I remind the audience that they can use that chat function under the Slido feature to enter their questions, and they'll be relayed to us. I'm not seeing any in my chat room right now, so I have a comment. First of all, the Mesa technology that you featured prominently went from basically soup to nuts in the last 9 months. And you might highlight that Thermo Fisher, a global technology holding company, just bought Mesa based on the RADx acceleration for $550 million last week. I don't know how many X valuation multiple that was, but that's an example of huge federal leveraging for a small, tiny company in California that went big in 9 months as an example of incredible translation. So my question following that, not seeing something in the chat, is that this is unprecedented times where translation, innovation, and movement to market under desperate situations can be accelerated, and the FDA's arrangement of their emergency use authorization has produced a lower barrier, so to speak, for entry to market, where basically a lot of things are being field tested. Even despite clinical validation and laboratory validation, there's a lot to be learned in how these things are being actually disseminated in the real world. Can you comment about validation this way, as an unprecedented way of us, United States at least, taking brand new technology and disseminating it rapidly for urgent reasons, but under a lower FDA validation criteria?
Yeah, this is, I think, one of the key, as I mentioned kind of at the end there, the regulatory challenges. I would even go so far as to say that under pandemic conditions, really urgent conditions like this, we need to create even an alternate path that's different from EUA. EUA and the lowered bar has been important in helping move things a little more quickly, of course, but we have a huge backlog of technologies that are pending EUA, and we could go even faster. It would be, I think, helpful if we created a path that was not entirely dependent on FDA, where multiple agencies could come together and rethink what are the regulatory barriers that we do need to put in place, kind of a purpose-driven thinking about regulation in the time of a pandemic. And then, of course, for permanence, we are expecting from our investments the performance of these technologies to be sufficiently high, and we're evaluating them all independently so that they could get permanent approval through the 510K or the PMA process from FDA. But I think as effective as the EUA process has been, in a sense, to encourage more applicants, it may not be optimized for the time pressures and needs that we have in a pandemic setting. And I am hoping that people can rethink that and get different stakeholders involved in thinking through how we might accelerate that.
Yeah, we have Peter Marks coming up here in the next session. We better be careful what we say here. But nonetheless, I think we have a tendency to throw the regulatory officials under the bus every time there's a delay in our well-intended process of translation. I would be very wary of doing that. They have a role, they have a job. It's a very important job of protecting public safety. And I think that we then have to produce a handshake where we understand each other's mission and trying to get this to move. Orla, you have a question from the chat.
Yeah. David, I do want to make sure I'm not misunderstood on that. The FDA has been spectacular. They're just totally overloaded. You can see that there have been so many applications, and they need our help. They were not designed to deal with this type of crisis. So I think the creation of expanded pathways would be helpful to them, keeping in the spirit of what they are doing.
Yeah, I wasn't intending to say anything, malign you in any way. It's a very difficult situation, and we have to respect all the partners. Orla, quick question here before we move to the
Okay, one quick question. Were the funds released more quickly and more easily compared to other more relaxed times, presumably non-pandemic?
Yes, well, really perceptive question, because that is at the core of the success. If you've ever had an NIH grant, you know you typically don't get funds for at least 9 months, if not a year. And certainly contracts on the scale of 20 to as high as $80 million that we've awarded will take multiple years. So yes, that structure that we put in place, we built on that existing network, we expanded it with some nonprofits, we created new mechanisms of rapid funding. And even though we expanded the structure, we reduced the bureaucracy, which is another unexpected result of this whole effort.
All right. Well, we're out of time. I just want to thank you, Bruce, for your time for us, and a very, very busy time for you, I'm sure. And I really appreciate you sharing your wisdom and sharing your experience in this very unique time with us. Thank you, Bruce Tromberg from the NIBIB. So we will move then in transition to our next session. Yeah, thanks. Have a great day. We'll move to our next session, and that will be then the regulatory sciences for novel therapy from cells, devices, and apps. And we have 4 speakers. And just a reminder, the 4 speakers have uploaded their video presentations to the website for the meeting. The website for the meeting allows you access to view those in real time tomorrow, whenever you have time to do that. And this particular session then is to review the take-home messages from those 4 different speakers and their presentations. And so the format will be that we will ask each speaker to highlight the main take-home messages from their talk with a short take-home slide. We'll have then all 4 speakers go for 5 minutes discussing their relative contributions, and then we'll have a discussion section, and then this will terminate then at 25 minutes past the hour, wherever you are. So without further ado, we'll go then to our first. I'll just remind you, the 4 speakers are Dr. Setsu Hashimoto from Japan. She's calling in, it's about 1:30, I think, her time, 1:40 her time. Hope she has a martini in hand. Andrew Carr from the University of Oxford, Stefan Schreck from the European Commission, and Caroline Paunoff from the OECD in Paris. Welcome, everybody. I'm delighted to have you. Thank you very much for your joining, and thank you for your wonderful contributions. I've reviewed all of your talks. I know that Orla's reviewed all of your talks, and I hope that the audience is familiar with what's going on. So let's move to Dr. Hashimoto and your summary slide. And I think that there's summary slide sets. Can you see your summary slide? Do you know your take-home messages? We're working with the meeting organizers to produce a venue where the take-home slide is available for them to see. I can't see it, but I don't know if you can. Sets, there we go. Sets, can you hear me now?
Okay. Yes.
Thanks. All right.
Yes, I can.
So here's your 4 take-home messages. Why don't you go ahead and provide a few minutes summary of what you said and how it relates to this slide, and we can move. Go ahead.
Okay. Thank you, Dave. My name is Setsuko Hashimoto. I'm a president of the company Cellseed, which is developing regenerative medicine products. And then I would like to share with our experiences. And then we have done the clinical studies and so on. So from our experiences, my first take-home message is that the regenerative medicine, which is based on the living cells, is not the same as the conventional drugs. So currently, in most of the countries, the regulatory system is used to approve those cell-based products is under the same system of the conventional drugs. But from our experiences, regenerative medicine products are not the same as conventional drugs, which requires the bilateral collaboration of the clinical practices and the product supplier. So the efficacy especially is very much affected how it is, how the surgery is applied and how the product is delivered to the site, and also even the post-operational patient care. So those things has to be taken into consideration when you assess the efficacy of these regenerative medicine products. And also for this, usually the first research started in academia and then do some small scale clinical research, and then the technology will be transferred to industry. And in that case, close communication between academia and industry from the very early stage of the development is very important. Otherwise, sometimes you have to repeat some same thing on both sides. And so in order to avoid any overlap works, the clear division of roles between academia and industry to take the advantage of their respective strengths. That is, we also have to consider. And then from those experiences, it is also, I would like to point out, this is not a simple linear model, but we have to do the cycle of the translational research and also the reverse translational research from the experience in the clinic. You also have to want to go back to the basic research or do some optimization of the manufacturing. So we have to go around this cycle of the translational and reverse translational research. That was our experiences. Thank you.
I forgot to mention that She represents Cellseed in Japan, which is one of the first Japanese biotechs to go public in a IPO in Japan and is listed on the JASDAQ. And with the Cellseed engineering product, she has 30 years of commercial experience and one of the few female Japanese CEOs in Japanese biotech after obtaining her PhD at Heidelberg University in molecular biology in Germany. I think that we'll return to your slide. One of the things you missed that I want to return to is this idea that we need a new regulatory framework to understand this first point, that regenerative medicine isn't conventional drugs, and therefore the regulatory frameworks in many countries is wrong to facilitate translation. So let's come back to that in the discussion. Thank you very much.
Okay.
Our next,
Thank you.
Our next presenter is Andy Carr. Andy Carr has been in our previous Translate meetings in previous years. He's the Nuffield Professor of Orthopedic Surgery at the University of Oxford. And his research has resulted in important contributions to improving the quality of and outcomes of orthopedic surgery, including large-scale, multi-center, randomized control trials in orthopedics, which of course are essential for any kind of medical evidence to move things forward. He's the co-founder of the Royal College of Surgeons in England and a trial unit at the University of Oxford in 2012. He's also been involved in developing the Oxford knee, now implanted in over 1.5 million patients worldwide, and he's pioneered the development of a patient-reported outcome score for surgery, which has been adopted also worldwide. He's a member of the Academy of Medical Sciences in the UK, and I welcome him to the floor. So Andy, you can see, I think your take-home message is there. You want to go take us through those?
Yeah, thanks very much. So my particular interest is in devices, medical devices, particularly implantable devices. And we've seen an extraordinary explosion in both the number and technological complexity of these medical devices. But what we've also seen in some instances is a problem with both the evaluation of efficacy and the evaluation of safety. So we've seen problems with metal and metal hip replacements, with implantable meshes in the pelvis, particularly in women. And to some extent or another, these problems represent a failure of translation, a failure of taking what seems to be a really good idea and a really good innovation into humans and determining whether or not this new technology is fit for purpose. Now, these issues that have arisen with implantable devices have resulted in changes to the regulatory framework, particularly in Europe, and we're just sort of beginning to see the evolution of the consequences of the change from a medical device directive to a medical device regulation. And the next few years, I think, will allow us to see whether or not these changes are working. We've also seen some changes in the USA that are not quite so dramatic, but there have been changes. I guess the place I'm coming from is very much from the position of clinical evaluation, effective clinical evaluation. And with others, we've been working on proposing some new technology readiness levels at this interface, this translational interface, as we go into humans. Animal models are very often inadequate or not fit for purpose in terms of evaluating a surgical device. Surgical devices often have a huge component of their effectiveness related to the mechanism of delivery. How do we get this device into a human body? That requires surgical skill. It requires an environment which allows the device to be effectively and safely implanted. We need to evaluate that in humans in properly designed trials. The other component of a device, a surgical device particularly, is that it often changes during the translational process. So unlike a pharmaceutical where the formulation is fixed with a device, it often goes through many iterations. So we need adaptive designs that cope with those changes that are consequent upon learning and consequent upon experience. So very briefly, what we're proposing is a series of phases of trials which are fit for purpose for surgical implants and are necessarily different to drugs. First of all, establishing safety and feasibility. So is it possible technically? And then in an ideal setting with the best possible surgeons in the best possible hospitals, in a randomized trial design, does this new technology work? And then address the next question, which is, does it work in a real world setting? What happens when this device goes out into general use and is put in its hundreds of thousands or millions in some situations? We need trial designs that address efficacy and cost efficacy in that sort of, with those sorts of questions. And then finally, there must be some sort of mechanism for long-term surveillance. One of the tragic issues associated with metal on metal hips around the world was we didn't know who'd had them because there was no unique identifier. There was no means of recalling patients and their metal on metal hip because we had no record of it. So what I'm saying is device technology is fascinating, it's brilliant, it's evolving, but we need an evaluation system process that's fit for purpose if we are to translate effectively. Thanks, David.
Great. Thank you, Professor Carr. That was a great synopsis. So our next speaker in this session is Stefan Schreck. He's from the Santé and the European Union Commission. He spoke about a lot of things that were, I think, classic pigeonholes in the way that governments operate with universities, operate with individuals in translation. He can go through his synopsis slide just a moment. He's been working in public health in the European Commission in Luxembourg for most of his career. In 2008, he became the head of the health unit at Executive Agency of the Health and Consumers, EHC, with the responsibility for implementing the EU health program. In 2011, he was appointed as the head of the Health Information Unit at DG Sanco, with the responsibility for health information policy. And in February 2016 to 2020, he was the head of DG Santé, health program and chronic disease unit, dealing with policies for all non-communicable diseases and annual work programs in the European Union health program. And since December 2020, just last month, he's the advisor for the stakeholder relations in the Public Health Directorate of DG Santé. So Dr. Schreck, talk to us.
Yes. Thank you very much. Yeah, and thank you for inviting me. And I will actually talk about something else, not really apps or devices, but the health systems of our member states in the European Union as a whole, because we have 27 different member states, and in some cases there are even regional health systems rather than national health systems. And what we try to do is to improve them by helping them to apply new knowledge. And since 2016, we have actually adopted a quite new approach to do that. And because what we had in the past was rather, we really suffered from the silos which exist. Researchers talking to researchers, public health specialists talking to public health specialists, health system administrators to their kind. Yeah, and there was really no information flow of a useful kind between those creating knowledge and those who would decide about their application in health systems. So we tried to change that. And the first problem that we discovered was the information flows. There is a lot of information about new approaches and new knowledge available in databases. There in Europe, we have many databases of best practices. The research program of the European Union finances thousands of projects every year, and the results are in big databases. But that information is mainly interesting for other scientists also doing research, or for the administrators who have to decide whether a certain grant is paid or not in the end, but not for the people in the health systems deciding about whether a certain change is implemented or not. For those people, the information was just not there. And then also the information is in compartments. So there's either only scientific information, or there's information about cost benefit, or there's information about piloting, or there's information about regulatory aspects, but what you don't have is all this information combined. And that is one area we are working on. We try to shape information in such a way that those who are deciding about how member states' health systems are shaped decide how the information is presented to them. Well, and then I've heard this already today. We try to not to do what we did in the past. So often we try to, in the past, we try to identify a certain approach for a health system we considered good, and then we recommended to everybody to use it. These days, we try to ask member states specifically what their problems are and what sort of solutions do they need. And then we try to look for, in this wealth of information that's available, for that specific sort of information and the solution and the approach and the research results they need. Yes, and then that's maybe specific for the European Union. In the past, we often try to find solutions that fit for all 27 member states at the same time, but we have to realize that there are big differences between the health systems of our 27 member states. And now we are a bit more modest, and we are already quite happy if we find a solution that helps a smaller group of member states, and then we help them practically to implement it. And that's my point 4. The system which we have actually offers all sorts of incentives and all sorts of practical and financial support, but it's really quite complicated to navigate. And there we actually also try to find a way, and there we did not achieve a lot. That's more an ambition at the moment, to help those people who have a problem, and where we can see a solution to find the right instruments in the European Union framework to actually implement that solution. Yeah, and finally, what I would also like to mention is that we try to focus also on measuring the success of what we do. We have, for example, asked the OECD to evaluate projects that are supposed to transfer best practices from a country where this works well to other countries interested in their implementation. And the OECD has a high reputation of knowledge in the field of cost-benefit analysis of healthcare systems. And with their help, we try to find out whether, on the one hand, the new approach has been properly implemented in the receiving countries, so whether the transfer of knowledge worked. And secondly, also whether actually the new approach delivered the results which were expected. And so to see in the end if everybody is happy. Yes, I think I'll leave it here. Thank you very much.
Sorry. Thank you very much, Stefan, for that synopsis. I remind the audience that you can input your questions into the chat function and the Slido feature on your conference website page, right-hand panel down low on the page. And we'll be coming up to the question discussion section just after our next speaker, Karolina Paunoff. She's a senior economist and head of the Secretariat for the OECD Working Party on Innovation and Technology Policy, and the Directorate of Science and Technology and Innovation at the OECD. So very timely after Dr. Schreck's description of his engagement with the OECD. She previously conducted work on innovation for inclusive growth and national intellectual property rights systems in emerging economy. And she's well published. She has a BA and a master's
collaborations across disciplines, across sectors, across different boundaries. And we heard from the first presentation that really the one key issue is how do you then go from university to industry across the different sectors? How do you really make this as smooth as possible? And it often is an obstacle because, of course, what happens, a research institution has its own dynamic, a researcher has dynamics of operating from what is somebody operating in an industry and what is even also the public sector. And then when we talk about health, we also talk about a very important role that the public health system plays. And there's these different, and also what, of course, and so there's an interplay here between those different sectors. And that is always the challenge when you try and articulate innovation. You would like those sectors all to speak the same way. If you want this to be successful and innovation to be stimulated, you would like to have all of these sectors speak to each other in a smooth way and come to a common understanding and not pose obstacles for one or the other. And I think my argument would be simply to say that when you talk about digital technologies, in addition to all of this, you have more possibilities to articulate that type of innovation, but you also have more challenges because the kinds of collaborations you want to engage in are much harder. And when you talk about a lot of the potential that you have from digital, often when it is not realized, it is because you need different actors to all agree on a common way of proceeding, notably when it comes to providing access to data, to sharing data, to protecting data. And there's a lot of items that need to be addressed to make that happen at a national, at a regional, at a national, and even more so at an international level. And I'll leave it at that.
Thank you very much, Caroline. So let me start with you. We have some questions coming in. I'll go to Orla, my co-chair, in just a moment. But if we try to segue what you just said with Stefan Schreck before you, and also some messages from yesterday about digital data, digital health data use. And I know this is, Caroline, it's not your expertise, but this idea of creating data that can be used by different cultures. So to reach back to Stefan Schreck, the fact that he has this diverse constituency within the European Union, you have a diverse constituency within the OECD, the types of data offered around the world in these databases is enormously diverse. There's not even a common system. There's not a common format. There's not a common download format. There's not a common security features. There's not common privatization features or policies. So how do we approach this problem if we're going to focus on medical data for just Dr. Schreck and for this audience? How do we approach this problem of the diverse patient user rights and the diverse user privileges for the people who want to use the data for all of these different nations, organizations, healthcare professions, politicians, researchers? How do we start to get our mind around this whole thing?
Do you want me to start on this?
Yeah, it's a daunting question. I think it's a compelling issue. Yes. Sorry, go ahead.
Yes. I will not. Let me just give you a general sense. I think it's what you're saying. I think one part that is important is to first of all also try and distinguish by the nature and the types of data. What are these user data, so patient data? Are these data that are generated in university and research contexts? Are these generated in industry contexts? And I think from that perspective, that's also how they're being generated and what use can they have and in what format. And then thinking about the incentives that these different groups have in creating them or in having them used. And one way that we are approaching it at the OECD, which is very much a high level part, is to work around a set of recommendations. And we have a set of recommendations that have to do with publicly generated data. We have a set of recommendations. We're talking about how to deal with private data. What are some of the ways to actually do that work of getting to a common understanding across different countries as to how we want to see data and really facilitate both their generation. We want to facilitate that they keep being generated and that they can be shared for the purposes that all constituents see as helpful without them being detrimental. So I would start it there and then give over to.
Clearly, this is a topic for huge discussion. I'd like to ask Dr. Streeck to respond to that from the EU perspective.
Yes, this is indeed both a problem and a big challenge, but also a real opportunity to make better use of health data in Europe, in particular across borders. And that has been identified as a priority by the Commission. So in the new EU health program, which, by the way, is much bigger than the old one. Actually, the budget is about 10 times the budget that we had still last year. That's, of course, the result of COVID-19. This program, however, does not just focus on pandemics, but also the European health data space is a big priority now, not only in this program, but also in other EU programs. So there's clearly an ambition and an objective to make better use of health data in the member states. We are aware that, of course, also we have the European General Data Protection Regulation, GDPR, and that puts in place rather strict requirements for exchange of data, in particular sensitive personal data. And that means that everybody has to be very pragmatic and very imaginative. And I know that it is possible to find solutions for collection and use of health data also in the European Union, even across borders. For example, one example I would like to mention is the European reference networks for rare diseases, which are actually now able to exchange patient data across borders to reference centers in a couple of member states, also for patients from other member states. However, it was necessary to establish a separate IT system for this purpose, which then the clinicians can use in the hospitals. So there are first solutions clearly available, and it is a priority for the European Union.
Thank you. All right, Orla, we have two questions in the chat. What do you have?
Okay, so this is from Frank Wissing for Andrew Carr. Do you have any suggestions for adaptive regulatory design for medical devices that are undergoing further development?
Yes, I mean, it's not particularly different to adaptive designs for other treatments, but it does require, during the course of a trial where randomization has been occurring, and I think there's been a big problem with lack of randomization in device evaluation in the past, but where a new innovation comes in, rather than discarding everything that's gone before, you incorporate that new device into your ongoing trial with a further arm to the evaluation. So that's the type of adaptive design that we think is appropriate. You bolt on the new innovation, and it may well be not just the implant, it might be the delivery technology, for example, that's changed. You don't discard everything that's gone before. You put the new innovation in alongside the previous trial design.
And what about the use of surrogate markers in adaptive trial design? There's been this argument that
I think that's absolutely fine. I think it's going to be a case-specific response to that question, isn't it? Do you have surrogate markers that you can use? What are your outcomes of value? If you can use surrogates, that's just fine. Very often with devices, that's quite tricky.
Okay, Orla, you have a second question? Thank you.
Yep. There's a second question for Caroline Panov. Have you identified areas where costs rather increased in comparison to other non-digital technologies?
Costs in the innovation process? Is that
I think the question's focused where digital costs are higher than conventional costs in terms of taking an innovation forward.
Well, I think it's. I mean, one of the big issues that is really how do you get access to that input, which is the data part. I think that's a major hindrance. It's a major hindrance, especially to some of the small players. So what we have been seeing is that some in a number of sectors, it's the smaller players that are often hindered but to apply like machine learning processes, etc., because they don't have enough in terms of data available that they can readily use. And that's become a huge issue and a huge obstacle to what you would think is the cost that you don't have and let's say in a traditional kind of production sense. I think data is an important element there, where you have seen some costs that are higher to be really more successful in this field. There's other issues as well. There's also issues to do with capabilities. So if you see your sector being disrupted by this digital, so we have digital agriculture, for example, there's a lot of kind of new machinery. It's all about the expertise and the capabilities to leverage that these new tools and technologies. And if you don't have those capacities, then you're actually struggling. So it's not something where it's cheaper for everyone and everybody can engage in this game. There's quite a few important barriers in terms of skills, capabilities, and also access to some of the inputs, which is data of different types. So.
Great, thank you. Seth Sachimoto, let me return to you. I think that Japan represents a very interesting case because the regulatory environment for most healthcare is very conservative typically, but because of the Yamanaka Nobel Prize, I think cell culture has enjoyed an enormous renaissance in Japan, and the regulatory profile and the barriers for cell therapies have therefore been lowered because of the faith in the Yamanaka Nobel Prize in iPS cells. So if we return to your regulatory framework, I think Japan represents a unique situation and a unique model right now where conservative regulatory approach is in fact less than I thought for the rest of the world. And therefore, in regenerative medicine, what are the barriers that you're talking about for the regulatory framework? Take off your mic.
Yeah. Okay.
There we go. Okay.
Yeah. So Japanese government introduced 2 new laws in 2014 after the stimulation by Dr. Yamanaka's Nobel Prize. And so in order to facilitate cell-based products to be delivered to the patients, so they introduced new 2 laws. The one is a modification of the existing regulatory law for the drug approval of the product. So this is mainly intended for the industry use. So it has to go through the preclinical and the clinical studies and so on. So in the same way as a conventional drug. But under this new law, there is also introduced a new system, 2 things. One is that originally it was meant for the pharmaceuticals and then the medical devices. So they introduced a 3rd category of regenerative medicine. So that is one new thing. And the second new thing is that the time limited and then the conditional approval system. And most of the cases that it is not so easy to make a large scale of phase 3 study with a controlled study. So we can do the small scale clinical study. And then when the safety is confirmed and then the efficacy is assumed, the ministry can give a conditional approval with a time-limited approval so that the product can already be delivered to the patient. But the industry has to monitor every case and then report periodically. And then at a certain time of the period, they have to compile all the data and then apply for the full approval. So this is a very new system. And then they also introduced another new law. This is mainly for the academia research, that the medical, under the medical doctor's responsibility, you can organize the small-scale clinical research and then to do some proof of concept data. Of course, in order to run this study, you have to apply the plan and then to the review committee. and then you will get the approval to run the study. And then in this case, there are 3 levels of the studies, depending on the level of the risks. So that the iPS-based cell using study, this is a highest risk level. So and so on. So that means that the academic research can give some proof, and also during that clinical research, you can also see some difficulties or some bottlenecks to operate and to run the clinical study in the future. So this is a good opportunity to learn the real clinical situation before you go into the clinical study, which is all the parameters are fixed, so that the academic research result can be well influenced to the industry development. So this is a new system in Japan. So by now we have 10 approved products in the regenerative medicine. Thank you.
You featured those in your talk very nicely. Thank you. All right, we have a question in the chat, Orla. No, there you go. Go ahead. Sorry, repeat that. Just Stefan Schreck.
Apologize. It's Hans-Dieter Volk has a question for Stefan Schreck. What is the strategy of DJ Sante to facilitate translational hubs, a key tool to translate creative ideas?
So, as I said, I was talking about actually translation of new approaches in the health systems. And for that, what we try to do is to strengthen our network of contact points, which we had already in the past in the member states, which were advising everybody in the member states about the European Union Health Programme and what it can do for individuals and organizations in the member states. And we would like, actually, we are in the process of upgrading this network by financing it better than we did it in the past. And the objective is to actually create hubs in each of the member states, which would inform people in those member states, not just about the health program, but also about the European Union research program, about potential funding from other sources like the European Investment Bank, and also from other programs which are managed by member states like the European Social Fund or the European Regional Development Fund. So what we try to achieve there is having a system in place where people in the member states that want to make use of new knowledge which exists in other member states or implementation of new research results to have something like a one-stop shop. But that will take a while. That's a complicated endeavor because there are so many instruments available at European level. And ideally, of course, we would also like to inform people at the same time about what is available nationally. So that's our interpretation of Translation Hub.
Thank you, Stefan. I have a question for Andy Carr. In terms of your take-home lesson about long-term surveillance problems and the ability to see parallel studies longitudinally out many, many years, what's wrong with registries? What can we do to fix those or promote those or junk those, throw them away? What do we do?
I don't think there's anything wrong with registries. Indeed, I think what we're seeing increasingly with devices is that the best trials, the best designed trials and the most efficient trials for recruitment and for reporting results on time are those that are derived from ongoing registry and observational studies. So what we propose is that for a particular technology, from the very start, a surveillance program is put in place in a registry, and then you use that vehicle, that registry vehicle, to set up and establish your trials that you embed within the registry. So the registry is the source of the trial material.
Correct. So the Swedish knee registry, for example, serves that purpose fine, but we simply need to expand them.
Yeah, there are a number of joint replacement registries around the world now which are working very effectively, and which we're increasingly seeing nested trials within, and really providing some very rapid recruitment to the trials and rapid answers to the questions we're posing. So I'm in favor of them.
Great. We have just a few minutes left. We have one question in the chat. Orla, I don't know if you want to convey that, but...
Yes, it's from Hans-Dieter Vogt for Andrew Carr. In CGT, academia moves forward more rapidly in the translational pipeline, including first-in-human trials. What is its role in the development of devices?
It's different, but it's not that different. The principles are the same. We want to know whether or not the device is safe and is it effective. And I think particularly with a device, we want to know, is the effectiveness contextualized? So how much is it important that you have a level of skill, for example, or that you have a particular environment, which I think it makes devices slightly different. And we need to have designs of investigation that cope with modifications to the device over time, which are very, very common.
All right, a last question for Carolina. You mentioned market dynamics in your talk. Can you give us some examples of market dynamics in the cases of digital innovation?
Yes, so that relates a bit to an issue as to there are two conflicting issues regarding digital innovation. On the one hand, you have fewer costs because you can do so many things these days on a laptop, as we are all now experiencing in unfortunate ways. But so there's lots of things you can do via laptop. You can do activities, you can do a software program, and you can kind of entirely deliver that to the entire market. You don't need to, if you're in the completely digital age, you have there are a lot of fixed costs you don't have. So in principle, you would think that this can generate a revolution of startups and new dynamics and new players in an industry. At the same time as you have dynamics that go in the other direction, because you have some big players like which we all know and which have which are related to Google, Amazon, etc., who have all these things, but in addition to that, they have a huge database and know everything we do and everything we want to buy, and that seems to be quite a relevant thing for doing other things and putting things on top of services. And so that then, and then you have a whole range of startups that are actually not being built to try and be the next Facebook, to be the next Google, to be the next challenger, but they are actually waiting to be acquired by the big ones, because what the idea here is more like you're a company, you're kind of providing an idea, and that is, if that idea is good enough, then some of the big players who have some of these tools that you need, you have access to the databases, you have access to all kinds of other infrastructures, will take advantage of that in the market. So there's different dynamics here. On the one hand, this idea of a democratization of innovation, where really anybody can just start their own company, so to say. And on the other hand, you have this tension of that doesn't seem to be quite the case in all circumstances. And how things will play out depends on many things. It depends on the context that is being provided. It depends on how things are playing out. I would say the COVID-19 context is changing things again in a different dynamic. So who will prevail in this type of context is quite an interesting question. There's on the one hand the winner-take-all dynamic, on the other hand a constant challenging of what's the new dynamic. And it's an interesting debate, because we had that already back in the days when Microsoft, which is still one of the major companies, was out there and everybody was worried about Microsoft's monopoly. And so some people will say, well, that's, we're still dynamic, digital is still changing, we still get new players and things will be shifting up. There will be something coming after Facebook and we'll have a new world type of, and others were saying, well, no, maybe we now have the infrastructures that will be with us for the next year. So we have established players in some of those industries and the dynamics will go differently. So there's different takes at the same level, and the question is what type of market dynamics will give us the best levels of innovation as we go into the future, and what type of regulation are needed. And I would just say that it's not so easy as to say, oh, here's a monopolist, that company is a monopolist, and it should be, because you need to think in terms of, in this digital economy, just putting together data is just efficiency enhancing. So the fact that you have everything in one service, so to say, is useful to you as a consumer. It's useful to the company to offer you better services on other dimensions. So it's a tricky issue to see how do you get competition in a context where pooling of information is just incredibly useful from a perspective of developing innovations. So that's what I would say on that.
Wow, quite an answer. Thank you very much. That was very thorough. We're at the end of our time. I've been delighted with this session. Thank you so much to Setz Hashimoto, off to sleep with you, 2 a.m. in the morning, Andy Carr, Stefan Schreck, and Carolina Paunoff. Thank you very much. And Orla, thanks for the co-chair. Great session. I remind everybody that we have a brief break right now, and at 30 minutes past the hour, we rejoin to the next session on translational hubs with Jörg Schultz and Hans-Dieter Volk. Thank you, everybody.
around, not ahead. The peak is just a way to walk at the sun. Look around, not ahead. A peak is just a way to walk at the sun. The forces on a hill, manifested by what you perceive as wind. Look around, not ahead.
Good morning, good afternoon, good evening, wherever you are. I welcome you to the session Translational Hubs, Infrastructures to Boost Translation. My name is Jörg Schulz. I'm from the Technical University in Aachen, and my function here is as a member of the Permanent Commission for Clinical Science by the German Research Foundation. The idea for this session, Translational Hubs, Infrastructures to Boost Translation, came from a consensus manuscript by the DFG Commission for Clinical Science, guided by our host, Georg Duda, and was published in 2019. In this paper, he and his co-authors proposed that setup and funding of translational hubs to foster translation in medical research and make translation more efficacious. For this session, we have invited 4 experts that will represent different cultures and structures to boost translation. We are particularly interested to hear how much entrepreneurial excellence, innovation, and translation can be expected in modern academic settings. What is already there and what is still missing? I also welcome my co-chair, Hans-Dieter Volk, who was already introduced yesterday, and I think needs no further introduction. As in all sessions, the full talks have been already uploaded, and I really recommend to listen to them. They are all truly excellent. They are very different, as I said, from different angles of the perspective. We will start with Professor Susanne Herold. She is full professor for pulmonary infections at the University in Gießen and the German Lung Center, and adjunct professor at Northwestern University Feinberg School of Medicine in Chicago. Her projects aim at defining novel therapeutic targets for treatment to attenuate lung injury and to foster organ regeneration and severe lung infections. The title of her talk will be Translation in Times of COVID-19 and Beyond. Susanne, the floor is yours.
Yeah, thank you. I'm still missing my slide, but maybe I will just start. I would like to provide you with a story with my view on how I actually did a translational process from a bench finding towards phase 2, phase 3 clinical trials. And I actually I provided an example of one particular compound that we are now applying, both in patients with ARDS, the acute respiratory distress syndrome, so lung failure due to different infections, including viral infections such as influenza and COVID, and which is also used in COVID patients that suffer from severe COVID pneumonia to prevent mechanical ventilation in those patients. So is there a possibility to increase the slide? Because I cannot actually read it. Otherwise, I will open it on my screen. Okay, so and this is my summary slide. Actually, I would like to mention what was relevant and very helpful at our center at the University of Gießen Lung Center to foster translation. And I have to say that we are part of, or actually the leading center of the German Lung Center, one of the German health centers that have been established some years ago to foster translation and to bridge basic science towards translation. So what have been key elements and infrastructure that were relevant and in place to enforce translation in my particular case? First of all, I want to start with a very important point that Georg Duda and co-authors actually pointed out in a Science Translational Medicine paper in 2014, and that is that basic biomedical research is actually instrumental to drive the discovery engine. And I would really make the point that I think this was the most important thing to drive this, also in my case, because we had all the relevant preclinical lung and infectious disease models in place to do all that research. With 3 CRCs, Sonderforschungsbereiche, 3 collaborative research centers, 1 on RNA viruses, 1 on innate immunity of the lung together with the Charité in Berlin, and 1 on pulmonary hypertension, another important lung disease, and 1 clinical research group also funded by the DFG, which I'm happy to head, and this is called virus-induced lung injury. And then we have an excellence cluster, Cardiopulmonary Institute, and as already mentioned, are part of the German health centers. So I think fostering or driving basic science was really important, and collaboration in that part. Now, then we went for different repurposing approaches, and of course it was favorable that a phase 1 or different phase 1 trials were already in place or had been, or we were able to rapidly accomplish them for our new application route, because we focus on inhalative application of our compounds. And we were happy to have collaborations within the German Lung Center, the DZL, with affiliated institutions such as the Fraunhofer Institute, ITEM in Hanover. And then the important mindset for translation. I think this was also one important driver of our success, because we had a translational pipeline and a local culture of building private-public partnerships successfully established at our lung center, particularly for the disease pulmonary hypertension. And 5 of the currently approved therapies for pulmonary hypertension have actually been developed in Gießen, from bench to bedside, towards worldwide approval. So there is kind of a culture for that. And what is also important is that failure is also accepted, because many of our trials also failed. But this is something that happens in the process and should not retain you from doing a clinical trial. Then another important infrastructure was the so-called Transmit as a hub for patenting and technology transfer issues in the value creation chain that is established at our university and is particularly helpful in patenting issues. And they do very good advice and are rapid and very, very effective. And another point that I would like to state here is that it is very important to have a clinician scientist tradition with structured clinician scientist programs in place, and hopefully soon also an advanced clinician scientist program. And our programs are funded by the German Research Foundation, also by the Excellence Cluster, Cardiopulmonary Institute, and by the German Health Center, by the DZL. And why is that important? It's not only important to combine basic research and clinical research and already start with the let's say proper view on the medical need in your bench work, but I think it also gets more and more important to understand the more and more complex therapies that we apply to patients, particularly if we go for more personalized therapies. It's very important that also clinicians at the bedside have a certain biomedical expertise and know more pathomechanisms of the disease to understand what they do with their patients. And then another important structure was also provided by the DZL, and this is a national biobank for lung samples combined with a data warehouse on phenotypic data of these patients. And that allowed a bedside-to-bench approach for precision phenotyping and molecular characterization of the patients. And therefore, and I think this is really important, for better protocol design and for better readout definition if you design a protocol. And in my case, I'm focusing, as mentioned, on ARDS, lung failure. However, this is not really a disease, but more a collection of different heterogeneous syndromes. this is why industry said that ARDS is the graveyard of pharmaceutical industry. Many, many large ARDS trials have failed, and we all believe in the ARDS community that this is due to the fact that we look ahead at a heterogeneous sampling of different syndromes and not at a proper disease. So we need a better phenotyping of cohorts, and we can only do so if we go for if we go for a deeper phenotyping, which is actually made possible by biobanking and data warehouse structures. And then finally, there are clinical trial infrastructures provided by the DZL as another part of these requested translational hubs, with clinical trial funding rapidly available after withdrawal of financial support by the company that first funded us. Although this it's not full funding, but it's partial funding. And then there is the KKS, the Coordinating Center for Clinical Trials, which is extremely helpful in study design, in protocol submission, in communication with all the authorities, and later on then also in trial management and CRO. So you see there is a lot of DZL occurring here. So I think this was very helpful, but we can also discuss what is still what structures are still lacking and need to be improved and maybe provided by these famous translation hubs that we are talking about. All right, thank you very much, and I'm happy to take your questions.
Yes, thank you very much. We move forward to the next talk, and then we'll have discussions all together at the end. So the next speaker will be Andrei Terčik. He's the founding director of the Michael and Mary Sue Shannon Institute at the Mayo Clinic Center for Regenerative Medicine. He has an endowed Marriott Family Professorship. He's also director at the National Institute of Health Cardiovascular Program and chair and President's Discovery and chair of the President's Discovery Translation Scientific Advisory Board. His talk will be about practice-centric translation strategy. Andrei. Andrei, you probably are still muted.
Yes, I was just saying how delighted I am to join you all. And as a member of the Berlin Institute of Health, also would like to congratulate our colleagues in Berlin for an outstanding organization. I would like here just to share some of our experiences at the Mayo Clinic. And as you will see, many of the concepts that we're developing here are very similar to topics you have already heard in various centers and various geographies. What is central for Mayo Clinic is the unmet patient need, that's the starting point and the ending point of sort. Finding the solutions for unmet patient needs, the centrality of the patient will be prevailing throughout all of the thinking. And strategically, the institution defines, let's say, in the upcoming decade, what will be the main domains to focus in that regard. That provides a discipline in building technological and translational pipelines, and those can be driven either very organically by the innovators, but often they are also promoted by business dimensions, including obviously the market pool that we mentioned before during this meeting. But what is maybe unique to Mayo Clinic, there is a significant directed also oversight and strategy implemented into those pipelines. The pipelines then lead us to the production of products which enter into a supply chain step, which is, I think, is increasingly new for healthcare delivery system, where in addition to innovating and delivering care, they're also becoming hubs for supply of new products, diagnostics or therapeutics. The next key steps for us is ensuring that these pipelines then evolve into becoming true clinical service lines, in other words, care pathways. And for that, there are three key elements, the elements of ensuring that they are approved at the regulatory level, the elements of adoption are in place, and ultimately that they are accessible, in other words, affordable for patients. That are key three premises. The integration into practice is an important also element because many of these technologies are disruptive in nature, but that integration has to be seamless, not to disrupt the healthcare system, rather to enrich it. And ultimately, it advances, it enriches the model of care with new solution addressing the unmet patient needs. Obviously, this process is continuously iterative across all the steps and collectively, and can be further enhanced by a number of elements such as, let's say, generation at each of these steps of alternative revenue streams through entrepreneurial activity and evergreen concept of maintaining sustainability of these pipeline service line models. While it's built initially at the patient level, it clearly have the opportunity of scale up, especially in the digital era, in addressing needs of diverse populations, and ultimately more globally contributing to the shared global knowledge and the buildup of a shared innovator ecosystem, with emphasis that I think at this time we need to underscore of deploying truly equitable innovation that will enable care equity, especially for areas that are sometimes underprivileged into securing that access. So let me stop here and looking forward to our other contributors.
Yes, thank you very much for your nice view. I think we move on. And the next speaker will be Stephen Kern. He is Deputy Director of Quantitative Sciences at the Bill and Melinda Gates Foundation. He's a strong advocate of making research data always fair and sometimes open to improve the impact data can have towards the problem it was collected to address and beyond. Stephen will talk about essential characteristics for effective translational research. Stephen.
Great, thank you. So if you got a chance to see my talk, or if not, then I hope that you'll be able to go through that. One of the main things that I focused on was this idea of creating what we call drug accelerators. And drug accelerators are the version that the Bill and Melinda Gates Foundation uses as a way to create translational hubs, if you will. But I think it's important to think about the term hub perhaps a little bit differently. So hubs, in my mind, tend to evoke a centralized place with a lot of things that are coming out from them. I think actually, if we're going to be really successful in these efforts of translational research, we need networks of hubs or hubs of hubs, right? So we need a connection of lots of hubs together. And you heard, if you listened to Bruce Stromberg this morning, talk about the RADx project that he's been leading. That's very much a network of groups that are doing very specific things. And in a similar way, that's what we try to do with our drug accelerators. And it's important then that they're interconnected and that you have a system for being able to hand off information so that your team or your university or your area can focus on your strengths and then pass to the next collaborator who actually takes it from you and moves it forward. In that regard, then our second biggest take home is that we want to focus on impact. And impact can mean different things to different people. Impact in the academic world may be very different from the impact in terms of trying to treat patients or get solutions in the hands of people. And so I think recognizing that is key. And again, we heard a great example this morning in the RADx, where you have a company that's going from new idea to being sold for $500 million in 9 months. Okay, well, if that's the case, you don't have time to wait for your New England Journal of Medicine publication to hold up that process, right? And so I think one of the things that's really important in the translational work is that we're trying to accelerate things moving forward. And as you start to move forward, you start to whittle down and chop off some of the ideas that aren't working very well so that you can actually have the winners move forward and actually have impact. Importantly, and we heard a number of the investigators, who just, both Andre and Susanna, talk about translation as a two-way street. We also heard earlier, I think it was Tetsuko, also talk about what she called translation moving in a reverse order, so from Tetsuko Hashimoto. So I think, working in the way that goes not just from bench to bedside, but actually also then feeds back bedside to bench, so we can check our assumptions, make sure that a lot of the work that we might have done preclinically to help build us and form us in the right direction is actually validated in the clinic and then fed back to say, okay, here's what we reinforced or here's what didn't work out, how do we move forward? So that two-way street is really key. And importantly, if you're going to have that two-way street, it has to be in the place where you need to work. So for the Gates Foundation, we actually are focusing a lot in low-income countries. So we need those translational centers to not be in Boston or London or Berlin. Nothing wrong with those. Those are great centers and good places to work with. But some of them have to be in low-income countries. They have to be in Dar es Salaam. They have to be in Nairobi, or they have to be in Cape Town, so that actually we can interface directly with the populations that are trying to benefit from that work. Fourth, you heard a number of people talk about mindset, and I think mindset and mentoring are absolutely critical for success. In my presentation, I talked about Kelly Chibale, who's leading a drug development center at the University of Cape Town, where he really incorporates this mindset of trying to move things forward. And that means that at some point in time, we have to kill some of the projects that maybe Kelly thinks are the most important or came out of his lab. But importantly, there's still other projects to go forward. Maybe they came from other investigators. So having that right mindset, and what, yesterday, if you listened, you heard my colleague Andrew Morris, who I'm working with on data platforms, talk about moving from an ecosystem to an ecosystem. And I think that was a really important distinction. It's a difficult thing because in academics, we're really rewarded and pushed for our individual contribution. And now we step into this translational space where perhaps our individual contribution has a different means. And we're part of a team, and then recognizing how we contribute and get success out of that is important. And that involves a really strong mindset difference. And helpful in that is having a mentoring group, people perhaps beyond just your immediate academic collaborators, but also people from outside the academic domains that are in the business world, that are in the policy world, that are in the government world, that actually have to implement. Or we heard, really good conversation with the regulatories and regulators to really make them part of this network so they can be advising and guiding the direction. And then last, for us as funders, as the Gates Foundation and many other organizations that are providing funding, we have to take this, what I call an investor mindset. So the investor mindset is one where we're actually going to be much more involved, much more hands-on and integral to the work as it's going forward, helping to provide insight and guidance, perhaps than we might otherwise as a traditional funder, where we'd just be catalyzing work and seeing what results come out of it. So I think if we can follow those 5 steps together very effectively, then we have a good setup for being able to do effective translational research. And I'll stop there. Thanks.
Thank you very much, Stephen, for your view of a community-based ecosystem. And then now we move to Professor Wolfgang Wick. He is chairman and professor of neurology at Heidelberg University and division head for neuro-oncology at the German Cancer Center. He has a translational research focus on precision medicine, on treatment resistance and immunotherapy, and he is spokesperson of Collaborative Research Center funded by the German Research Foundation. Wolfgang will talk about translating novel concepts into clinical application. Wolfgang.
Yeah, thank you, Jörg. I already learned a lot from all of my colleagues and took some notes, and just to focus a little bit on a very specialized topic in medicine, it's neuro-oncology, is about brain tumors. My view is, and although I understood ego and eco, that it starts with excellent innovative science, and this may even be ego-driven. So I think really concepts that are different from what we heard before. I have a few examples in my presentation. One is about brain tumor networks, which just changed the view we looked at brain tumors. It's an unsolved problem for decades, and I think this change of view is a prerequisite for a successful translation. And I think this is matching what you said before. If we are talking about translation, we need dedicated funding structures, and I think Susanne talked on that as well. So it's not only about the project, it's about the people at different levels. It's about clinicians, clinician scientists, probably advanced clinician scientists. It's about the trial. It's about what comes after the trial. So we need the whole chain funded, not just individual steps. And this is necessary, and this oftentimes is lacking on one spot. So if we are talking about translation, we need to ensure that we have really tested on each step that we have the appropriate funding structures in place. What we learned here, also for the brain tumor field, but I think this is true also for others, is that translation, if we talk about things that are moving from our labs to bedside, it's potentially not the main problem. It's transfer. It's bringing this into an industrial, into a community application, making this a product, but also thinking about how this is influencing society, how this is influencing the way that jobs are generated or other people are just, things are done a little better with that. So I think this is important, and not just focus on translation. Collaboration is helpful to shape the focus, and as we learned, for example, with the DFG supported SFB, it's very interesting to implement a translational chain into individual projects. So have the whole structure, not only as we all do, at the end comes translation, but within each individual project to have the translation already built in and make every investigator think about that concept, that what he or she is doing is really necessary to translate to something at the end. And then if successful, if that is selected, if that is one path, also following a transfer path. We learned that specifically if you're working with different institutions, and the German health centers have not solved that in total, we need a harmonized legal and support framework. So this is oftentimes the biggest hurdle for successful translation or transfer, that even on one campus, if you have different institutions, if you have different legal entities, you're stuck with the next step. This necessitates one inflexible point of contact. I think this cannot be underestimated. And of course, starting in neuro-oncology with a very, very small example, with something really specialized, probably not anyone here in the audience interested in, I think you need to make that an example of a bigger structure. What we are doing at the campus in Heidelberg now is understanding that we have a lot of excellent topics around us. We have a certain infrastructure that is really successful, both in the ego and in the ecosystem, but really combining that under a common umbrella. And this umbrella, I think, first of all, needs to be content. So starting with the first point again, excellent innovative science. With that, I'd like to stop, and I'm grateful for any questions. Thank you.
Yes, thank you very much, all of you, for your nice presentations. I think Susanne and Wolfgang made it really clear that there must be strong basic science then to accelerate to translational research. But we would like to define here what is needed to make that possible, what can be done in academia for what is industry, and a good collaboration there needed. And you also made clear that there are sometimes gaps that make it very complicated to do that. And you need the ecosystem to go ahead. So if you had a wish list for the infrastructure to foster translational medicine or transfer, Wolfgang, what you said, we have a little different definition what transfer is. So what would be on your wish list? And maybe Susanne starts and then it goes to Wolfgang.
Yes, there's a lot of things on the wish list, but maybe I start with the ones that are most important for me. What I think is lacking is an early start of education in translation and transfer. So we have a lot of graduate programs. We have programs for MD students that want to go into science, even at our university. But what I realized, and what was my own situation as well, was that when I came to the point where I should do the step towards maybe a phase 1 or a phase 2, so now we have all these nice animal data, and now we should go, I was totally, I didn't know what to do and what are the next steps. So I was happy that I had good mentors in place and all these good structures that helped me, but I was not aware of anything that I needed now to start and to implement and how the processes are. And if I would have known this earlier, maybe my basic science projects would have been designed a little different, to already think of what do authorities need and which data do you need to generate in addition. So I think that it would be good to start flanking educational programs in clinician scientist programs, in graduate programs, to start earlier to let these people know what is important in this process. So I think education is one important point. And then, of course, funding structures in Germany is also another point. I mean, we have the BMBF and we have the DZGs, but for the larger clinical trials, this is never sufficient. So if you do not have good PPPs, like private-public partnerships in place, or if you do not have these industry contacts, and sometimes this is just the case, if you enter into a new area of research or whatever, then funding is an issue. And I think we can improve here. And it's not only funding for the study directly, but also funding for translational hubs as requested by the DFG in its position paper. Maybe I stop here and maybe others have other points of their wishes.
Yeah, for me, I fully support the educational aspect. I think building this into what we are doing early on, I think is important. It did not work so far on our campus. I would come back to this complete workflow. So I think for me, checking this on every aspect, so it starts with the basic science and then with the modeling, which helps you to ensure that this is real, and then some medical chemistry and some lead compound identification, getting back to the model and getting back basically to the basic science and then forwarding it to the clinic. So really having a full chain and making this irrespective of the whether it's neuro-oncology or cardiovascular or infectious disease. I think this would be for me something which I think is needed, and I don't see that in the German, at least in the German surrounding, that there is a full flow for this, and this could start somewhere, and then this should be a model and also in collaboration done for many centers. So it's, I think, not something which needs to be established in every center. This could be done in multiple centers, and I think the German health institutes, the DZGs, could be a very nice starting point for this.
Andre, one question to you. I mean, you have set up a prime example at the Mayo Clinic in Germany. That's what Susanne and Wolfgang discussed. We have mostly project fundings, but we have difficulties to fund like these research infrastructures. And my question is about sustainability. So can you set up something and then it can sustain itself? What would be the recommendation from your experience? I think you are muted.
Sustainability is a critical element. I think my colleagues highlighted already this concept of continuum. So building this continuum, we call it discovery, translation, application, has to have the iterative component which is linked to the evergreen sustainable element. Healthcare providers typically look at the clinical practice as the main financial output, but actually the alternative revenue streams that can come at any steps of the continuum are very critical. And Susanna highlighted the education, and in other words, the build out of the specialized workforce of the future is an absolute catalyst and an absolute must in that evergreen process. And including the next generation is essential in the sustainability aspect long term. So we have built at Mayo not only the programs themselves, which we have built not as ivory towers, in other words, isolated institutes, but we have embedded them in each of the specialties. So we then let the specialties really drive the innovation based on their needs. And then ultimately it's a collective success. So the unison of forces is essentially what allows the matching of the innovation and the scientific drive with the ultimate healthcare output and addressing the unmet needs of patients.
Thank you very much. Steve, you made the point of collaborative success versus individual success. What are the incentives? I mean, it's very easy to envision what incentives are for collaborative success, as we heard today morning on COVID-19 or in your presentation on tuberculosis and so on. But of course, we have the difficulties with the young people who also need their individual success. So how can we implement that?
I think what you're hearing is actually an interconnectivity among all of these topics that all of our, the speakers have presented. And to this point that I think if funders can have the patience to say, we're funding a network, not necessarily a specific project, or a collaboration and a group of people that are working together on a problem, then the solutions can come out of that network. That network helps support it. You can provide training and teaching programs to get people into the right mindset, but if the students don't see that there's some downstream consequence of them doing that, they're not going to go in that direction, right? So they respond very quickly to the opportunities, I think, that are present. So you have to create that ecosystem that allows people to succeed. Now, once you have that, there are still components, and that's one of the things that I've loved about our drug accelerators. We have some very, traditional academic researchers who are participating in that. They have a role that they contribute. They have a broader role, and they use that as a way to leverage their broader role of what they do in their own research outside of the accelerator. So I think there's a place for both. It doesn't have to be an either or. But the key is that if you put these structures in place, you have to be willing and patient to allow them to grow and to build for impact. Because if you don't, then people are afraid to kill projects. They're afraid to basically fail. And that's the only way you're going to learn is if you say, okay, I'm going to fail. That's an accepted component. That's actually helping us know that we're moving towards the right solutions instead of continuing to work on the wrong solutions and have something that's going to be productive in the long run.
Okay, I have one more important question, but I will put that in later because there are questions in the Slido, and so I hand over to Hans-Dieter.
Yeah, okay. Thank you, Jörg. I think there are a couple of questions, but maybe before we go to the questions, maybe a short analysis of the live poll. The question was, have you ever taken advantage of translational infrastructure at your academic institutes? And 22 people answered. And the good news are 68%, as the majority say yes. That means this is coming more and more popular that we have translational centers. Only 23% say no, and 9% I'm not sure. Okay, coming to the questions which came in. The first one is from Frank Wissing regarding translational hubs. And he is asking Susan, what kind of expertise and resources are needed on local levels versus or what could be better provided on national or even international level? And I think that's a good question, and maybe it's also a question to Andre. Okay, maybe Susan can start.
Maybe I start. With respect to the translational hubs, as they had been identified or described by the DFG, initially it was not meant to have a hub at a certain university, but more or less to have hubs all over Germany, which integrate the different academic centers. So I think a very important thing is that, and this has already been mentioned by Wolfgang and others, that we build up better networks and structures in the academic settings. Because in my particular case, actually it was that I asked my friends and colleagues whom I knew and who I know, okay, they would maybe be part of my trial, but they said, sometimes they say yes because they know you and say, okay, I like what you do and so on. But it's all dependent on your personal networking. And I think we need better structures to provide, let's say, to give you the chance to immediately and easier create or have a network where you can implement trials very rapidly. And I think this would be one of the major parts of such translational hubs, that they would enforce and enable these networks and set up larger infrastructures that combine academic centers in Germany, but eventually also beyond. And maybe I can just tell from my recent experience in COVID trials, which should run worldwide, how difficult it was to harmonize protocols over different countries. So I think this is also something, particularly in cases of a pandemic, where we really need to move forward and get better harmonization of what do the different authorities want and how. I mean, currently it's very different, right? And it was very, very difficult to do that. So this would also be maybe a task of such translational hubs to maybe get this better set up and done in future and be a hub or a crystallization point between the researchers and the academic field and the authorities, and not only locally but also globally or at least European wide. So yeah, maybe this. I hope this answers Frank Wissing's question.
I think those are good points. I think maybe I would follow with your idea also not to think only on the local and national level, but also international level. I think that I completely agree with this. And we tried it in the cell and gene therapy to form international, as a European-wide network, the flagship initiative or Restore. But I see it's not so easy. Of course, we have two words. On one hand, we have the competition. On the other hand, the need for collaboration. And we have to bring this together. And the second point is there is missing funding for this kind of international structures. There are some elements in the European Union funds, but that is still limited. From my opinion, it should be more. What is your opinion? Maybe particularly the European part, guys, here?
Yeah, maybe I just add on this. Yes, I mean, we made up our mind just with this COVID experience. So in this particular case, it's about the Solidarity, the large WHO trial, where it was really, really difficult to get protocols that are that were designed to run in all over the countries and like globally to get this approved in Germany for good reasons, I have to say. But we have to find measures and structures where we can fund, let's say, networks or structures that help with that. So, and as you say, there is no funding for such European-wide or even worldwide structures to do that. I think particularly in case of pandemics or something like that, it's really important to get this.
Okay, thanks, Susan. Other comments from others people?
Yeah. Sorry.
I was going to say, I mean, one other thing that I'd add in is that, for instance, with the European FP projects that had come out, so I think it was the last call in some of the EU projects, we were able to incorporate a group from EU funding into the tuberculosis accelerator. So they actually took their work and then were able to complement it with our other international work. And in a similar way, you know, if you're trying to develop new therapeutics for tuberculosis or for malaria, or for any immune functioning project, it has a huge overlap with oncology. It has a huge overlap with respiratory care. And so there's always opportunities to be able to do some of the fundamental things the same. And so that's where I think, understanding what other networks are out there and how your experience that might be in a very particular area from a translation of preclinical to clinical could be extended into other areas outside of the disease area where you happen to focus.
Okay, thanks. Dr. Wick, you have a comment?
Yeah, it was just echoing on this, because this is, of course, also important for the rare diseases. So it's important for pandemics, but also for the rare diseases, and potentially the rare diseases where at the end, money in a very small surrounding is not the big issue, and where potentially, since the disease is so difficult, money should potentially not be the biggest issue. And there, I think international collaboration in making this happen would be even more attractive. We manage quite well in the brain tumor area, but on the other hand, if it comes to medication, money is getting into the way. And I think this is also getting into the way of successful transfers sometimes. So I would be interested in Stephen's opinion on that, knowing that the foundation is trying to overcome those kind of obstructions.
Yeah, and particularly one of the things is to be proactive. And I think you maybe even mentioned it, Wolfgang, in your talk. And Susanna mentioned it as a challenge. She saw it looking downstream and saying, well, what's going to be the next thing, right? So it might be fine that I can get something into a phase 3 trial. But if it's a technology that's going to be way too expensive for anyone to be able to use, then it's going to have a solution that doesn't necessarily have impact. So being able to think about that in the front end. And one of the things I talked about in my presentation was just anticipating cost being an issue and then trying to solve it very early, so you don't solve it later on and then have to repeat all of your clinical trials again because you've changed the structure of whatever you've done by reducing the cost. So that's where I think having that end-to-end perspective, really a key component of the translational centers and the translational mindset to have good impact.
One way to look at this is this ultimate maybe drive to achieve translation without borders. Whether you take it geographically or structurally, I think that's ultimately the collective mission. The local structure usually brings very specialized expertise. And then the ability to move them nationally, internationally, globally enables also to bring in public structures, public health, sociology domains, socioeconomic domains, and so on. The beautiful examples come actually outside of biomedicine. Let's say the NASA project, bringing the man to the moon or beyond, I think gives us examples how collectively we can achieve this concept of translation without borders, which I think it's an essential driver for the future.
Okay, thanks, Andre. There's one more question to Stephen Kahn from Frank Wissing. Translation is about taking risks. How do we facilitate the switch from academic to industrial political risk culture during translation? Question to Stephen.
Yeah, so I mean, one of the ways that we've, especially following on the comment that was just made about rare diseases, so partly as funders, if we say this is part of our strategy and this is part of our objective, then we're going to incentivize that development or maybe support it in a number of ways that would be very atypical. And so, in essence, perhaps create a market for something that otherwise doesn't have a market, so that actually facilitates the translation. But that's not, it's a unique situation. And I think that's one of the challenges is thinking through, okay, well, where are there other options where I can perhaps piggyback this technology onto a use that will make revenue and support a sustainable business, but then still be able to peel off and work in an otherwise non-sustainable space. So this idea of dual use, very common for what we're thinking about. Oftentimes, the example that I use is a project where we're developing it for a long-acting therapeutic for malaria, but its primary indication is going to be in schizophrenia, and being able to do weekly dosing of medications in schizophrenia. Again, a small market, but these things all built together start to create a sustainable platform that then one entity can follow on. So I think it's having that creativity of putting the blocks together in ways that we don't normally think about that help us get over some of those hurdles.
Okay, the time is almost over, but we have one interesting question. Maybe we cannot discuss it completely, and also we have not all people on board here. The question by Andrew Morris: the Innovation Medicine Initiative, the IMI program of Europe, has spent more than €5 billion on more than 160 projects in the last couple of years. The question is, has it delivered any impact, and what would you change about it? I have to say I'm very critical to this program, but maybe there are other meanings or comments to this point from our speakers here. Okay.
Well, I will say that the IMI program, that was the one actually that did link into our TB drug accelerator. So we did see that it was able to actually leverage a clinical trial opportunity to start doing early phase screening of TB compounds and down select things that weren't working. So there was an example of that. But I think challenge we run into with the bigger entities like IMI is that they are incredibly bureaucratic. And so you're spending a lot of funding on just the transfer between institutions. We talked about just, sometimes even between departments or institutes in the same institution or within the same country. And now we're going to go on 27 countries, and then we're going to get globally. So we have to think about ways that we can make a streamlined connection that's going to make this feasible. Or, academics and scientists who are motivated by science, not by contracting, are not going to put up with it.
I completely agree. And also my experience from academic point of view is with the IMI programs, it's also not an open game between the different pharma companies and academic members, because there's too much secrets between, and therefore I think there's still a lot of space for improvement. Unfortunately, we have no time to discuss it because the session is over. I give it back to my chairman.
Yes, time is over. Thank you very much, all of the speakers, for the excellent contributions and the questions. It was a great session, and we only have 4 or 5 minutes break, and then we'll continue with the program. Thanks a lot, guys.
around, not ahead. A peak is just a way to walk at the sun. The forces on a hill manifested by what you perceive as wind. Look around, not ahead. Look around, not ahead. The peak is just a way to walk at the sun.
Yeah, good morning, good afternoon, good evening, wherever you are. Welcome to this next session on the topic accelerated translation in times of global crisis. I think that is a very actual topic because of the situation everybody knows. I think we have excellent speakers on board. Sami Aberman from Pluristem, Peter Marx from the FDA, Katja Bose from the Jens Zuckerberg Initiative, and Klaus Tschitschek from the Paul Ehrlich Institute in Germany. That means we have different aspects, regulators, non-for-profit organizations, biotech companies. I think is a very broad view. And I can only invite you to watch the pre-recorded talks as for all sessions. It's really worth to watch. Here we start now with a short summary, short statements And the first speaker, first of all, I'd like to introduce also my co-chairman. I think it's not necessary to explain more. Both of us, Georg Duda and myself, were introduced yesterday. Okay, let's start with the first speaker, Sami Ammerman. He serves as the executive chairman of the board of Pluristem, an Israel-based company, biotech company, joined in 2005 and redirected in the company's strategy towards cellular therapeutics to become one of the global leaders in this industry. Sami Ammerman visions to use the maternal portion of the placenta as a cell source for Pluristem's product, combined with Pluristem's special, unique 3D culturing technology, led to the development of the company's unique therapies. In 2013, the World Stem Cell Regenerative Medicine Congress ranked Dr. Ammerman as one of the 50 most influential people in the stem cell research. I'm happy that we have you here. Sami, the stage is yours.
Thank you very much. Just to let you know that we missed most of your introduction because there was a noise from the system, so nobody can hear you, so I hope it's recorded. So thank you for the opportunity. And we at Pluristem, we are manufacturing a product from the placenta. All our products are derived from the placenta, and the rationale behind that is to fulfill our mission to convert the miracle of birth to therapeutic for all. And we have 7 clinical programs as we speak, and we expect to have 3 clinical data readout in the next 12 months. One of the unique features of our company that from day one, we made the decision to invest heavily in manufacturing and not to start phase 3 studies before we have a full-scale in-house GMP certified advanced manufacturing facility. And we are very proud on that because we are unique from that purpose in the industry, because we make that decision because we understood that cell therapy is completely different from other drugs development, antibodies or so, because cells are sensitive creatures and they adopt their secretion profile and properties based on the culture conditions they are facing. And because of that, we made a decision not to enter into phase 3 studies without an approved manufacturing facility with the required QA, QC, qualification method, and so on. And we are very happy to work with the FDA and the European College Institute and the EMA by collaborating with them and improving the manufacturing processes before the phase 3 studies. We believe that in order to do a good translational research, you have to have a deep understanding of the mechanism of action of your cells, and on the other end, the disease prognosis and what can influence the disease, and that's exactly what we've tried to do in the beginning of the pandemic, that we moved our activities not only for ischemia and muscle regeneration, but also to treat COVID-19. And we were happy to be approved by the PI and by the FDA to start phase 2 clinical studies based on the safety profile that we generated in our other studies with the PLX-PD product. A key for translational research is collaboration. We are very proud in our initial collaboration starting in 2007 with the Charité of Berlin. And you can see the list below the slide, hopefully you can see it. It looks like very small, but we have a variety of collaboration with the Charité, with the NIAID, with the BARDA, with NASA, and additional entities. And we see that if you want to do translational, good translational research, you have to have collaboration with a variety of entities that have its contribution to the result that we expect to achieve. Thank you very much.
Yeah, thank you very much, Sami, for this summary. Okay, we move forward to the next speaker, Peter Marks. He is director of the Center for Biologics Evaluation and Research, the so-called CBER at the FDA. The center is responsible for assuring the safety and efficacy of biological products, including vaccines, allergenic products, blood and blood products, and cellular, tissue, and gene therapies, a new category of therapies. Dr. Marks and center staff are committed to facilitate the development of biologic products and providing oversight throughout the product lifecycle. I think there are a lot of examples, and we are very happy to look forward. And please, it's for you. Peter, do you hear me?
He's muted.
Peter, you are muted.
I think he's not speaking. He's not muted. He may not be able to hear you.
But you hear me, yeah?
Yeah, we hear you.
Okay. Peter, do you hear me?
I think he may not realize that you're that he's on stage. I think he's on the phone.
Peter? Yeah, I think we move forward to the next speaker and go back later. The next is Katja Brose from the Chan Zuckerberg Initiative. She is a science program officer at this initiative. The goals of Chan Zuckerberg Science Initiative are to support basic science and technology that will make it possible to cure, prevent, or managing all diseases by the end of the century. That is a high aim. Before joining, she was part of the editorial team at Cell Press for almost 20 years, where from 2004 to 2017, she was the editor-in-chief of Neuron and publishing director at Cell Press Elsevier. She has been an active member at the Society for Neuroscience as a member of the Professional Development and Neuroscience Training Committees. She earned her undergraduate degree in the Brown University with double concentration in biology and European history, a very special combination. And she received her PhD in biochemistry from UCSF. Okay, I think we are very happy to have you here. Katja, it's your stage.
Okay, thank you. Thank you for including me in this interesting panel. The first thing I'd say, which I said in my talk, is I feel a bit of the odd man, woman out here because I don't work on translational research or clinical research or COVID for that matter. I'm a program officer at the Chan Zuckerberg Initiative. And as was mentioned, we're a relatively new philanthropy. We're about 5 years old now, and focused on 3 initiatives: education, justice and opportunity, so an advocacy mission, and science. And within the science initiative, our focus is very much on basic science as a foundation towards clinical and translational impact. And so what I had put onto that summary slide for you today, and you can hear more about in my talk, is really kind of more a series of questions than answers in thinking about how this past year with the pandemic, and I think also if one thinks globally, sort of a period of a lot of socio-political and economic unrest, what can we learn about that will strengthen the scientific community, the whole of the scientific ecosystem, certainly basic science, but also translational clinical science. And at CZI, we're quite engaged in developing kind of new tools and opportunities to promote interdisciplinary science, collaboration, and open science as tools to accelerate and improve science. And I think, in fact, the COVID pandemic has really kind of reinforced to us how valid those tools are. So when one thinks about the progress that was made this year on understanding the biology of the virus, and as well as the pace of accelerating vaccine development, there's no doubt that the rapid pace of discovery and progress would not have happened without essentially decades of investment in basic science. You know, likewise, we've seen during the pandemic increasing collaboration, open science, sharing of results and data early, the rise of preprints and open data sharing, open sharing of health data. These are all things that I think we've been trying to incentivize within the broader community, and the pandemic has done it for us. And so the question that I leave this panel with is really kind of how can we harness what we learned during the pandemic and not let it go after the pandemic is in our rearview mirror, if you will. So I think those are some of the positive things that have come out of the pandemic. Again, interdisciplinary science, more collaboration, more open science, and speed of discovery. There have been really disruptive effects as well. And I think we should not forget those, and we should not paper over them as we move forward. 3 actually that I highlighted in my talk were, this is focused on, I think, sort of my perspective on basic science, but I think it is also true throughout the ecosystem. The first is thinking about the workforce of science. So who does the science? A disproportionate impact of the crisis on female researchers, especially women with children, and young early career next generation scientists. So whether it's early professors in the academic infrastructure, or whether it's postdocs. I mean, there's really been a difficult impact of the crisis. And again, some of this political social unrest, especially that we've been seeing in the US on this next generation. And I think if this isn't attended to, we will, I think it's probably an exaggeration to see that we'll lose a generation of scientists, but it won't be far off from that. I think really thinking about how this pandemic has impacted different parts of the population is going to be really important. Likewise, I think the pandemic has really shed light on issues of disparities and inequities in our healthcare systems. Certainly, this is true in the US. I mean, you have better healthcare systems, I think, in terms of sort of global access in Europe. But I would say that disparities and inequities exist worldwide. We only have to look at the differences between the global north and the global south. This, too, I think, is an issue that I think the pandemic has put into sharp light, and we shouldn't forget it when the pandemic recedes into history. I think it's important to also say that science is front of stage again, and this period has reinforced the value that science and technology have for society. We can see this obviously with the hopefully the benefits that the vaccine will bring to reopening society. But I do think that it's also a time to take stock of how society fits into what we do as scientists and those of us in the translational and clinical communities. The research community needs to really kind of lead on these issues. I think bringing more social scientists into the field, thinking about differences in the way that we measure success and impact and new incentives and rewards. And importantly, making sure that science benefits all of society. Again, not losing sight of some of the inequities that the pandemic has really kind of brought to bear on our society. And I will stop there. Again, my point was mainly to raise those questions. Are we going to, as a field, as an ecosystem, get over the crisis and then move on and return to where we were? Or will we use this time to really take stock and maybe reinvent how science and society intersect? Thank you very much.
Yeah, thank you very much, Katja. And we move forward. Peter, do you hear me now? Peter? Okay, then we move forward to Klaus Cziczek. He is a biochemist and extraordinary professor of biochemistry at the University of Frankfurt am Main in Germany. He took over in 1999, deputy director of the Paul Institute, and from 2001 to 2009, he was vice president of the Paul Institute, the regulatory authority in Germany, particularly for biologics and cell and gene therapy. And since December 2009, he has been the president of the Paul Institute. Since February 2014, he has been member of the WHO Product Development for Vaccines Advisory Committee. And Professor Cziczek is the author of more than 150 publications in international scientific journals. And I think your organization was in the focus in the last couple of weeks because of the new vaccines. It's great to have you here. We listen to you.
Thank you very much. I hope you can hear me well and also see me. It's a pleasure to be with you. And I can say that the COVID vaccine will certainly keep us busy for more than this year. And that's true for all of us, I guess.
Now we lost you. Do you hear him? Hello? Dr. Cziczek, we lost you. We cannot hear you. Did you switch off the headset? How can we solve the problem through technique? Okay, you get a call in a minute. Sorry for for these problems. Okay. Und hat uns auch nicht, ne, Peter, ne? Ja. It's working again? Dr. Chichek? Can you hear us, the others? Yeah. Sami,
Yeah,
Katja, you also can hear me?
I can absolutely hear you. I
Okay,
think
that
his
means we have
headphones
only a
are working,
problem.
and
We have only a problem to listen to Frankfurt. I don't know why. I think you are back. No. No. Disappeared again. Okay, then let's make a short break. We try to finish it and to solve it. And we start with the questions which came up first to Sami. No? I hand over to my co-moderator.
Yeah, thank you very much for your introduction, Sami. The two questions that came are from Dave Granger. How to assess or assert preclinical model therapeutic validation with human allogenic cell products beyond an immune incompetent mouse that lacks disease aspects. I'm pretty sure that's an easy one for you, Sami. And if you could give a little bit of an insight, and probably also in the perspective of, did you need to show any of that in the COVID perspective? Would be probably my add-on for that. Sami.
No, we don't hear you. Wow. Now we have a real
No, it's my mistake. No, it was my mistake.
Yes. Okay, it's fine.
Sorry.
Yes. Sami, you are back.
Thank you for the question. Yeah. Thank you for the question. Do you want me to continue or do you want to continue the session? Sorry.
No, please, Sami,
yes,
answer.
I know that. Yeah.
Okay. I will try to answer. Dieter, please stop me.
Peter Marx, I think we also hear you now. No, it's gone. Sami, I think it's good if you could simply stick to your...
Okay. We are in the allogeneic space.
Können Sie...
As you know, there is not sufficient animal model to deal with the immunological interaction between the cells, MSC-like cells, and the human immune system.
Yes, hopefully you can hear me.
Yeah, we can hear you, but now we have Sami is on.
So probably, Dieter, they will continue with the presentation and I will answer later because we are...
Okay, but Peter, do you hear us? Peter, do you hear us? No. Wow, that's a crazy session.
That's a crazy
It's the first one. Sorry about this. It's very hard to continue.
Hello?
Yeah, now we hear you.
Now we hear from time to time. We hear you.
Can you hear me?
Yes.
Yes. Okay, great.
Okay, then I introduce you. Peter Marx, he is the director.
Okay, tschüss.
Okay, maybe I make a short introduction of Peter Marx. He is the director of the Center for Biologics Evaluation Research at the FDA. I have done it before, and we are happy that you have me now on board. Peter, you can start if you hear me. Please, it's your stage. Peter? You are muted. Peter, please speak.
Hello?
Yeah,
Hello?
Yeah, we hear you. We hear you. Could you...
Why is not continue?
Yes. I think it's fixed now. It was, I think, a double muted thing. Es tut mir leid.
We hear you perfectly, Peter.
Ja, das geht. Alles Gutes. Vielen Dank. Es tut mir leid. Okay.
Please start. It's your talk.
Thank you. Okay, so thanks very much. So I want to be able to tell you today about some of the practices that we've had of accelerating product development and approval. Our take-home message here, obviously, is that there are a number of best practices that may continue, such as early communication with regulatory agencies, integrating different phases into one clinical trial. But another an important piece of this accelerating product development is that we have learned the importance of adequate and agile manufacturing capacity because that has really been very clear to us. The next slide, please. Okay.
We can see your take-home message.
Okay.
Slides.
Okay. You know what? I won't worry about that. Let me just rather than worrying about the slides, I'll just talk to the issues for a few minutes. Just to be clear, we at FDA have tried our best to have conversations with regulatory agencies early on between sponsors and us to be able to have these dialogues very early on when people are first starting to put together their development plans. So this has made a big difference, particularly in the time of COVID-19, where rather than doing a lot of development work, people have come to speak to the agency before even starting phase 1, sometimes even before doing a lot of preclinical work so that they can understand what we're looking for. This has allowed us to help certain products fall out of development more quickly. And it's also helped some products make it into development more quickly. So this dialogue, I think, has been very helpful. We've continued that dialogue as products progress through phases 1, 2, and 3 of development. And that's been a benefit to us in part because as we have, for instance, with the vaccines, taken them to emergency use authorization, our way of making them available during this pandemic without an actual license, we've been very knowledgeable about the data. And this concept in the United States, which has been particularly important, of integrating different phases into one clinical trial has been quite important because particularly in the United States, our clinical trial system is one that goes in fits and starts. When people do phase 1 and are completing that, there is downtime of a matter of months between that trial and the next trial, and the same thing between phase 2 and phase 3. So the goal here is to try to avoid that problem by having things go seamlessly. And I'll stop there because I think we wanted to have time for discussion at the end.
Yeah, thank you, Peter, for this short summary. And I'd like to hand over now to Peter Cichohek again. Can we hear you? Yeah. Peter, can we hear you?
Nee, ist nicht zu hören.
No, we don't. We cannot hear you. No, sorry.
Call, ruft, glaube ich, an. Ja, verstanden.
Okay, then the technical support will try to find a solution. Then we can now continue with question and answers. Sorry for this a little bit confusion, but it's the only way we can do. I hand over to Georg again.
I'm trying to come back to the question that we briefly touched with Sami. We have the question from Dave Granger that you're using your cells. It's a xeno model. If you have that in preclinical trial, how can you verify? How could you convince authorities like FDA and EMA or Paul Ehrlich Institute to get the approval for your cells? Also now in the perspective, probably, Sami, if you could add that in the perspective of the COVID pandemic, because apparently there is not an adequate model system preclinically available.
Okay, so thank you for the opportunity to present our approach. We start very carefully to treat patients back in 2009. We treated 3-by-3 cohort of patients looking into the immunogenicity issues. And then we confirmed together with the Charité the unique immunological properties of the cells and actually immune privilege properties when injected into humans. When we came to the COVID-19, we already had about 500 patients that have been treated with our product. And therefore, we have been delighted by having the approval of the FDA first at Paul Ehrlich Institute. Second, based on the fact that we have proven clinical data that show non-issue with immunogenicities. And second, we had a manufacturing facility that had been reviewed by the agencies for phase 3 studies and have been confirmed that we are good enough to move forward to phase 3 and marketing later on. I hope I answered your question.
Yeah, thank you very much, Sami, and I think head over to Dieter.
Okay, I've heard from the technical support that Klaus is back. We can hear you now. Can you try, please? No.
1, 2, 1, 2, 1, 2.
Yeah,
perfect.
Great.
Perfect. Here we go. Okay, let me give it another try. So,
finally,
you introduced me already. Yeah.
Okay.
Thanks very much. Yeah, you introduced me fine. So let me just try to introduce what we are doing in terms of translational science. So Paul Ehrlich Institute, as I tried to explain, is similar to US FDA CBER. And we basically parallel our regulatory support through the whole development process, starting from drug discovery through clinical trial authorizations, of course, support for inspections for the manufacturing. And also at the licensing stage, we do the assessments in the committee of the European Medicines Agency. And beyond that, we're involved in pharmacovigilance and, of course, experimental batch testing and batch release government-wise. Now, why are we committed to support translational science? Simply, I think it's very important to start early on to fill the development pipeline. And I think access to regulatory authorities has always been a little bit difficult for, first of all, academics who are involved in translational research, but second of all, also for small biotech companies. And as we explained earlier, now that we're living in times where COVID vaccine development, for example, is very important. And we've seen that a rather small biotech company like BioNTech, now teaming up with Pfizer, was able to bring a vaccine product to the market. I think that shows that our support has worked trying to engage with these kind of developers early on. Where's our experience from? Well, first of all, we opened up many years ago what is called an innovation office. That's simply a telephone number and an email address for early access of academics and biotech industry to the Paul Ehrlich Institute, and for being able to also allow them to ask very simple questions that are down to the regulatory basics in the beginning. Second of all, I think we also have experience because we are committed to do research, experimental research in the laboratory and with animals ourselves. Of course, staying away from real product development. We also form the basis and have ideas, produce scientific ideas for concepts for new medicines or new vaccines. And with that, our heads of the product-specific divisions, of course, are connected to the different fields of immunology, virology, microbiology, allergology, cell and gene therapy, and so on and so on. And that gives us early access. In addition to that, since a while, we had collaborations with what we call in Germany, German Health Research Centers, which of course, in terms of project support by the European Commission, exists also on the European scale. And here we have an example of what is called a product development unit. This product development unit has a commercial part that's apart from the Paul Ehrlich Institute, but has an office for scientific and regulatory advice at the Paul Ehrlich Institute. And here we are in the middle of the research and also the early developments that are done by academics, and it's very important to help them early on with scientific advice. Now, let me come to the summary and to the success factors according to our experience in translational research. I think it is important that contributors of translational research have to synergize, for example, in such translational research centers. And of course, giving these examples that are specific for development research in infectiology or in cancer or in lung and other things, one could think of bringing all that together centrally in Germany, maybe centrally in Europe. Second of all, I think very important is to combine the experiences of the different fields. There is the basic academic research that is very important, that produces great new ideas and concepts for medicines to be developed. There is also the development expertise in commercial ways from the biotech and the pharma industry. That's very important because, to our experience, very often academics think rather complicated in details and miss sometimes the basics they have to do just for the first clinical trial they have to do. Apart from that, of course, also the finances are very often not provided by usual grants. And then the third part, the regulatory knowledge and expertise. And in Germany and Europe, there have been attempts, for example, to educate academics better in regulatory knowledge. In our experience, it's much better to turn to the professionals, for example, of the Paul Ehrlich Institute, to use them very early on. But of course, the terminology sometimes is different, but due to the fact that we are committed to work with the scientific fields, that has improved quite a bit. And then I think it's important to realize that there are certain points where handover to other players in the field is very important. That is, from the basic and translational research hand over to those that have clinical trial centers at the university clinics, to those that provide, for example, GLP facilities for the non-clinical investigations, and third of all, to use service providers, for example, for the GMP manufacture of the medicines to be developed, of the vaccines to be developed to allow first use in humans. And then, of course, a very important handover is also from, let's say, academics with early clinical trials or biotech with early clinical trials to hand over to experienced large companies in the field that know how to go through and carry product development through to licensing and beyond. And one other additional fact we have experienced is to be acknowledged. That is the fact that academics very often cling to their kind of research ideas they have developed once upon a time, and because they were not able to test them in clinical trials. And sometimes they like to also ignore rather, let's say, sobering experiences when it comes to first clinical trial testing. It's important to have a group that sets clear milestones, controls them, and develops termination criteria for development projects, because it's very important. Many projects are excellent and will be carried out, and through to licensing, some other projects may just be too early to be realized, and that's an experience we all share. So in summary, I think there are three factors that make things go for development: basic research, including university clinics, regulatory knowledge and commercial development knowledge. And that's the way how we propose to proceed to the German government, because also the usual scientific project funding that, that goes maybe for 3 years or 5 years is just not long enough for an extended need for development that's all sometimes extends to 10 years or so. With that, I want to finish. Thank you for your attention. And I'm very happy and glad that finally I could be heard and this works. Thank you.
Thank you, Klaus, for this talk. And I think we hand over now to the questions. And there's one more to Katja. And
probably, I must admit, I enjoyed all 4 of your talks to listen to them, and I enjoyed seeing them. And I have tons of questions. But I'd like to specifically point out a question that Jochen Büchel brought to the podium that goes to the perspective that Katja Rose took, sort of the a little bit unusual or different perspective, probably looking into what is it actually that sort of challenged also the scientific system in the current mode. And the question is, Katja, is diversity for you also related to knowledge areas? And I understand it in the way, is it probably specifically in medicine where we have a number of life science projects that have lack of an impact, or where we have probably a higher risk that projects sometimes don't hit and really impact, like probably in other disciplines where there is a faster evaluation or an easier mode of analysis if the impact can actually be reached. So in your view into this, have you seen any diversity between different scientific fields, or would you say no, this is basically pretty common across sciences?
Yeah, hopefully I understand the question well. I mean, I do think that diverse approaches are important to solve problems generally, scientific problems included. And as we think about our strategy at CZI, so at CZI, for instance, I lead our program on neurodegenerative disease. And a key mission of the work that we do on neurodegeneration is, one, to really kind of reconceptualize how we think about neurodegenerative disease for a more diverse perspective. So, for instance, a lot of neurodegenerative diseases, like a lot of diseases, are very much studied in these siloed communities. And there's a lot to be learned by looking beyond the silos. And so, researchers in Parkinson's disease can learn something from researchers in ALS or Alzheimer's disease, and there needs to be kind of more flow between fields. I think also bringing interdisciplinary approaches to bear will naturally kind of diversify a field in terms of scientific perspective. And so we see this a lot in neurodegenerative disease that I'm trained as a neuroscientist. We neuroscientists tend to think in terms of neurons, but there's been an increasing appreciation that, of course, the nervous system is part of the larger body, and that, inflammatory processes, metabolic processes, the influence of aging, development, and life experience all have effects on brain function. And that's true for COVID too. I mean, we're seeing that so many of these, we call them in the US sort of pre-existing conditions, sort of diabetes, cardiovascular issues, kind of influence the larger state of the health of a person and make them, more or less susceptible to, the dramatic impacts that COVID can have. So in terms of, I think, how we think about this as scientists, I think we just need more people looking at these problems with a diversity of approaches. And then, of course, those approaches will have different timescales. I mean, it's these days a lot easier to do, for instance, a genomics or a proteomics or kind of an omics experiment that it might be to establish a new model system, or really looking at the trajectory, say, of aging on any of these processes. So, of course, I think we need that kind of diversity. I think when I was talking about diversity in my talk, I was speaking more from the perspective of who does the science and the kind of questions that who does the science may illuminate. I think there's been good studies in the social sciences that scientists from different backgrounds, different racial, ethnic, geographic backgrounds will study problems in a different way. We'll choose different problems to study. And I think that too is important, that we not get too caught up in kind of one way of even conceptualizing the problem. Hopefully that answers the question.
No, thank you very much.
Okay, thank you. Maybe I have a question to Peter and Klaus regarding the regulations this particular way. I think it was very impressive to observe in the last couple of months how fast the regulators on both sides of the ocean and also in other countries like UK or Israel, how fast they get approval for clinical trials, but even more approval of new drugs, particularly for vaccines. And I think, of course, there was a high pressure by politics and society, but the question is really, what can we learn? What can you learn as regulators from this? Is this a special situation in the pandemic environment, or do you think you can also learn for the future, for a normal life, that the approval of trials and drug approvals, marketing authorization might be faster by new tools?
You want me to start?
Yes, you can.
Go ahead, Klaus.
Okay. Peter, sorry for this. I didn't know you could switch on. Anyway, I think we made several experiences, and these are very worthy to be taken over. First of all, I think in terms of pandemics, we learn a lot how to deal with them, also in terms of product development, including vaccines, and we'll take that over and see that we can be better prepared also in the regulatory and development field to the next pandemic. And tools like new vaccine platforms and things like those very much help. Number two, we learned and we experienced that it's very great to work in harmony in the world. So not only the WHO, but also the International Coalition of Medicines Regulatory Authorities allowed some kind of harmonizing discussions along the way, and basically the vaccine development on the regulatory side, also the biomedicines development, was supported by allowing some preclinical studies to be really done preclinical, others to be run parallel to clinical trials, and to combine phases of clinical trials that we normally had one after the other. And of course, administratively, there's the rolling review. But to answer your question that you initially also had, I'd like to say that when you look at different product developments, of course, they take some time, and I'm not sure whether we can take the experience over in a way to say, okay, suddenly we'll develop all vaccines within a year. From our regulatory side, that is no problem, but it needs synergy between larger experienced industry, small biotechs and academics, as I explained. And as you see from the developments from Moderna and some other things, very clearly this helps to combine knowledge of different fields, and I think that will help also in the future. And to come back to the question, is it more difficult in some fields to develop things than in other fields? We have the experience that it depends also on the way how a product is designed. We are regulating biomedicine, so I'm not talking about the small molecules work. But if you look at the biomedicines, the experience has it, there are platforms, for example, like monoclonal antibodies that can be readily used, are easy to manufacture, and you need ideas how to tackle a certain disease and how to pull up a certain mechanism. Then there are things like cell and gene therapy, where it sometimes is very difficult to find the right way, the right genetics to tackle a disease. Then there are things like allergology, where you need, for example, a new idea now, like taking immunotherapy to a different stage, and that's much more difficult. So I think depending on the technology and the different fields, it's more or less difficult to develop no product. And then at the end, I want to say, I would wish that all the products that could help patients and vaccinees could be developed in no time, but it needs also an enormous effort from the side of industry, from the side of academics, an enormous amount of finances. And this has been provided in this situation, and that will not be there afterwards. Absolutely. And I think we just have to find that we can take some experiences over, learn from them, but other things won't go as easy because there's no pressure. At the end, there's one word for regulatory licensing that holds also in this and future situations. That's a positive benefit risk balance of products, which means if there's a lot of risk and there's a lot of pandemics, maybe less than normal benefits could also do it in some instances, but in a normal situation, that's not the case, and that's another lesson we can learn over.
Okay, thanks, Klaus. Peter, some additional comments to this topic?
You know, I largely agree with Klaus. I would say that our major learning here are essentially is essentially trying to reduce time by condensing certain steps, avoiding dead space between trials, and having an active dialogue between the developer and the regulator. Pretty much those are the key things I think we can take out of this. There are some things that are not going to continue after the pandemic, but those I hope will. Over.
Probably just from a slightly different perspective, and as a question to all of you. Again, Pristwich asked the question, in our translational educational processes, how do we teach about and thus minimize confirmation bias? So how much of what we have now learned as lessons should we integrate even much earlier into educational programs? And I don't know who would like to start, probably Katja, from your perspective, if you want to, but I think everybody has a lot of expertise to answer from your perspective.
Yeah, I mean, going back to my journal editor days, I mean, I think it's not just about confirmation bias. It's about all issues around rigor, reproducibility, and bias that I think have to be educated from earliest stages. I don't actually think it's only a training issue. I think often people are well trained, and then I think, as somebody was kind of alluding to, the sort of incentives or pressures or forces within the processes get people who know well otherwise to bend. So I think it needs kind of consistent and persistent attention, but certainly training is one piece of that.
And probably before the others answer, would you think this is actually the risk is higher if you say it's not training, it's the pressure? If in such a pressure time when everybody is seeking for solutions, is that not the moment when this is even at higher risk? I don't know how the regulatory side see that with Peter and Klaus or Sami.
Peter, you start.
Peter? Do you have any comment to this?
Yeah. So, I think that we're in a place where we've made the mistakes that have been made particularly in the United States. Bias has led us down the wrong path enough times that we are, I think, cautioned that this is something that we have to train all our scientists in very early on. To avoid going down the path of making the mistakes that we've made over the past year during this pandemic, because we've made several that are really, they're almost beginners' mistakes, because people rush to do things without thinking through them or using their training.
Yeah, maybe I can chip in by saying I think it's a question of training. That's one aspect. But I think we just also have to learn that less positive results may happen, and we have to accept them. Yeah. And that's fine. And many people think, I have a great idea. I come now with a clinical trial. It worked in the mouse, and now it has to happen in humans. That's not always the case, and that's how life works. So I think one thinking that Peter and I have also promoted is the iterative going into clinical trial, coming back to the bench, trying again. This kind of thing simply happens, and not everything will work like a charm. We are very lucky that actually the COVID vaccines work and seem to work excellently in some cases. We couldn't have guessed so, and in some instances, your suitable animal model just is not good enough to show that something will work in humans. And that's something that has nothing to do with training, it's just experience of life, and we'll go through it over and over again and try hard.
I mean, I think also this is a place
learning
where
acceptance. It's acceptance. I mean, unfortunately, it's people get so enamored of their child that they don't understand that it may not be able to become the brilliant scientist that it may want it to be.
That's what I meant when I pointed out on my slide, we need decision criteria. And we need people to stop sometimes developments, particularly in the academic era in Germany and sometimes in Europe. What has happened is that people stick to their ideas and publish great papers for 10 years. But because of lack of funding, lack of opportunities, they have never been able to carry that, for example, into a clinical trial. And once it happens, they cannot accept that it did not work. And I think we just have to say we have to learn from trials in humans, and that's the end of the story. There it works or it doesn't work.
Sami, any
I
comments from
think this
you?
is also a place where collaboration and where open science can and should help. I mean, having the ability to incentivize replication studies from the very beginning to just have more people, I think, testing common models will give us more data and more, hopefully, robust data across multiple labs and institutes. So I certainly agree with all points made, but wanted to just iterate that I don't think it's just the training issue. I think it's about the long term.
Okay, Sami, do you have any comments from the biotech view?
Yes, I think that it's important to mention that to go first to clinic make a difference. And we luckily have been approved by the Israeli Ministry of Health to start compassionate use treatment in the beginning of the COVID-19 pandemic, which gave us initial data that we have been approached later on. We approached later on to the FDA and the PI, and then we had some evidence that it's working. It's about the pandemic issue, about the preclinical studies. As an habit, what we do the same animal models in 2 different labs in order to ensure that if the results are the same, then we can move on. Because sometimes you have a very, very successful animal model in lab A and not necessarily in lab B, and then you have some question mark that you have to resolve before you move on.
If I may chip into the conversation, it starts to get interesting. Now, I think compassionate use is really a wonderful thing to help patients in a desperate situation. That's fine. And the pandemic may have some aspects that need for compassionate use. But in general, I think our dogma is that the clinical trial setting is the right one to safeguard the patients, and it's the right one to get good evidence for anything. And, sometimes this happens that doctors and developers come to us and they had a couple of compassionate uses that look quite good, but when it comes to a real evidence-based clinical trial, the whole picture falls apart. So at Paul Ehrlich Institute, we do not promote the idea that compassionate use is a good preparation for a clinical trial. Just to make sure that you all understand that.
Okay, we completely agree. But one comment on that. As you know, there are many diseases that you don't have an appropriate animal model to prove anything. So somehow you have to bridge a gap between your feeling animal models and the understanding that you may help patients. And the compassionate use sometimes is a good proof of concept. It's not a clinical study. We do not accept them as any evidence to efficacy and so on, but it gives you a feeling that you are not dreaming on something that cannot be fulfilled.
Much better is a small clinical trial with 10 patients only.
Okay,
yeah, but
I think that is an endless discussion. I think is very, very important and very interesting. Unfortunately, the time is over. We have added some time because of the technical problems, but anyway, now it's over. Thank you very much for this very interesting and lively discussion. Enjoy the evening or the day. Bye-bye.
Yeah, thank you very much for your contributions, and we're continuing now with the consensus paper draft 1, and we directly jump into this. Dieter, would you start, or is Glenn Prestwich on?
Glenn should do it. I hope he is there.
I hope Glenn is already with us, and Brenna.
Looks like they need a short break, no? Glenn is there, I see it.
No, Glenn is there and Brenna is there. Perfect.
Perfect.
We're here. We have to turn off the live feed.
Oh, good.
So Glenn, would you take over and introduce the discussion? We have the idea to come back to the 2 drafts of the consensus paper papers. We would start with consensus paper draft 1, measuring translation, what works, what does not. After the presentation yesterday, the discussions that came afterwards, do you have, how would you like to pitch? Do you have questions that you would like to bring in here directly, or what would you propose from your side?
Well, what I would propose is I've prepared, with Brenna and I have prepared 3 polls. The first of the polls is on translational activities at your institution. And the question that we've posed in the first poll, which you should be able to see, I hope, now while we're starting, is the following. Are translational activities currently considered in your evaluations for faculty promotion and tenure at your institution? So this is about where you are specifically, not generically, but are they strongly supported, a little bit, neutral, somewhat discouraged, or strongly discouraged? And I know from personal experience that we can be anywhere on that spectrum. Brenna?
I've seen all universities. I think there's trade-offs with supporting translation within P&T decisions and how you incorporate that input. So I'll be interested to see the responses.
Haha, very interesting. So far, what I'm seeing is 60%, 50% somewhat, 33% neutral, and 17% somewhat discouraged. So no strong feelings on either side from academic institutions. They're kind of wavering somewhere in the middle to supportive. And that's not unsurprising. That's kind of what I've seen. I think if we'd done this 5-10 years ago, we would have seen a much bigger shift towards discouragement. Now I'm starting to see some discouragement come in. So while this is going on, I'm going to just introduce the rest of what we're doing. The next question that you'll see when we start talking about this, our effort here with these polls is to create data that we can put in a form of a graphic or a table in the consensus paper. And if you've seen the paper draft itself, you'll understand where the next poll question is coming from, which will be on the screen in about another minute or so, which it concerns the most important translational metrics. We had a list in the draft manuscript. We've added a few, subtracted a few. Those will be coming up next. The third thing that we'll see will be addressing the axes of impact based on that figure for translation, the landscape of translational impact that we showed. And that will be the third poll, but we'll get to that a little bit later. So if I could ask the technical folks to stop the current poll, capture all that data, store it, so that we have, now that people have responded to the live poll on translational support, now we're going to go to the live poll on what are the most important translational metrics. And this is going to take a little while for people to fill out. We want you to pick 5. I think there were 12 or 13 or maybe even 14 in this list, depending on what we finally ended up with. But there are a lot of different possibilities for what you think as a participant ought to be measured at your institution, whether it's a government institute, a university, what have you. Brenda, do you have any comments about the ones that we pulled up and highlighted?
So I think one of the things to consider when taking a look at these metrics is which metrics are relevant depend greatly on where you are in the ecosystem and translational pipeline. So invention disclosures may be a useful metric for, say, a technology transfer office, but is that the right metric for judging translation? So I think each of the metrics should be taken within the context and the stakeholders that are being evaluated, and as part of the holistic perspective across the axis of impact that we're taking a look at.
Yeah, and this is very much comparing apples, oranges, rutabagas, and train rides. So some of these don't really connect with each other. particularly well. But this is what we are faced with when I've walked into a dean's office and they say, well, we're all supportive of translational activities. How do we measure them? What are you going to count? And so the concept of bean counting is the hardest thing when trying to be reduced to practice to put into an academic constraint. But we have to realize, as we heard from several of the speakers in the last three sessions, that not every translational impact is measured differently in different entities, whether it's an academic institution, a company, a government office, your grandmother's living room. There are all different ways to measure translational impact. So we're coming up with some very rough estimates of this, and that's what we're trying to get at in what to measure, what not to measure, and how to measure it. So your input is going to be very important to this. And as I'm talking, I have to send my answer here. So what I'm seeing, which is very interesting, is there's a lot of support for societal impact, TRLs, technology readiness, patents, startups, and industry contract right now. Contract research are the top 5 right now, but these are we're now watching them change in real time. It's kind of exciting to watch these thermometers go up and down. But we're still at the highest levels for societal impact and technology readiness with patents and contract research with industry and disclosures coming up close to startup companies. So we're seeing a general sense that revenue generation, paper citations, publications, economic impact, licenses aren't as important as some of these other things. I think this is going to be very interesting for us to put forward in the consensus paper to kind of get away from what I call confirmation bias. I know that when I go into something like this, I know what I think is going to happen, and it's really interesting to see what actually does happen. Brenna, you've probably experienced confirmation bias all the time in your investing.
Yes, and I think within the context of measuring translation as well, it's important to keep in mind that what gets measured, what matters is what gets measured, and or vice versa. So as we look at the metrics, we reify them in some ways and start comparing across over time. We need to make sure that those metrics are the most appropriate ones, that we're asking the right questions of those metrics, and that we're incentivizing the behaviors and processes and efficiencies that we'd like to see in advancing translation. So I think the role of the metrics, as well as how we put them in context, again, are important for impacting incentives and measuring progress over time.
Yeah, I think the most important aspect, as you pointed out very clearly, was you get what you reward. And so if you want people to do something, that's where you put your prize money, your recognition, your academic awards, the things that advance you in promotion and tenure. Or if you're in industry, if the people who are recognized as employee of the month or drug discovery of the month of the year, those are the kinds of big scale and larger scale incentives that drive what goes on in academic and industry as well. So,
and I think as well, as you incentivize different parts of the value chain, you may be extracting resources from other stages as well. So as more disclosures come in the door, that's more effort and time and attention that's required to take a look at those innovations and advance them. So prioritizing in each stage and the shape of that funnel and the shape of the metrics, I think is a relevant consideration as well in talking about this.
Excellent. Well, this is. I think what we'll do is we'll call a pause on this particular question on the metrics. Things have sort of stabilized now, and let's go to the next one. The next question we'll capture that. That will be in the consensus paper in some form. The next thing are what are the most important axes of impact in the actual diagram that we presented in the draft. We have the number of individuals impacted as the x-axis. The y-axis was individual health impact, and the z-axis was overall societal value impact. But other things that we proposed in the figure legend, it could be something in return on investment in economic terms. It could be something having to do with disability adjusted life years, which was a suggestion from Maurizio Vecchione, who is now at the Terasaki Institute. It could be something other, and the particular poll doesn't allow us to have an other write-in, but you have a way to write it in. You have a way to email it or send it in the Q&A, and we can bring up, if you send it in the Q&A, we can definitely talk about those in real time as we come forward with this. Maybe we need a different landscape for each institutional entity type. You can suggest that as well. But I'm really excited to see what kind of feedback we get on this particular one, and we're looking for the other axes of impact, in which by which we could present, hopefully quantifiable in some way, aspects of 3 different axes so that you can see it on a page. Actually, as I mentioned before, these are really 4 or 5, 6, 8 multi-dimensional space that's just harder to visualize. So let's go ahead and see where we're at. So we're getting good. Top 3 are still the ones that are in that landscape. I call that confirmation bias. But we're also getting some feedback on ROI and daily dailies are as a possibility for including in the axis. So obviously some of those are more readily quantifiable than the ones that we originally listed. But maybe Brenna, if you want to comment on what you're seeing on the poll.
So one of the questions that comes to my mind from the poll and the question is, how do the axes of impact vary based on the mission of the organization or the lens that you use for taking a look at impact? So if you're looking at this from an economic development perspective, from an academic perspective, from an industry commercialization perspective, how do those vary, and where should you devote the efforts, and how do you measure the differences in those magnitudes? So I think incorporating the mission-driven aspect of it, I think, is an interesting question.
So people have been pretty much focused so far on the polls, and now I think that polling has gone. Actually, it's changing still. So I don't want to close this off, and I'm interested to see if the people who have put other in others. If you would like to type those into the Q&A, then that would be a great way for us to bring those other things forward and be able to look on the different lenses that are possible for different institutions.
I think I would like to add some aspects in the discussion because after finishing the poll, I think there was one discussion. We have one letter was written by Hans Limbus. He say, okay, we have to differentiate between lagging and leading metrics, also looking back and looking forward. I think that's an important point we can discuss. And the second point, I think, particularly for the young people, is, of course, it sounds very good and it's very, very important to have a high impact for the society and for the individual patient and so on. But that is a process that normally need 5-10 years or even more to bring it to visible, to see the impact. For the young people, of course, you need also interim step that they see some success and some progress to get motivation and incentives. That I think we should also not forget. That is a very complex situation, how to measure and who is measuring it.
Brennan, do you want to comment on the lagging and leading indicator question first?
So yes, I think on the lagging versus leading indicator, I think it also depends on where you're looking at it from the value chain. So for a measure of impact from an academic institution versus industry versus a government or national perspective, I think are all very different. The lagging indicator for one may be the leading indicator for the other. The point. For example, research funding and venture capital funding. So I think taking into account the perspective of the stakeholder, as well as considering which metrics are actually meaningful from a risk perspective. So at each stage, there's a drop off in a probability of success. And some of the early indicators are not reliable signals for later stage translational success. For example, only about 1 in 1,000 invention disclosures ultimately end up getting commercialized. So how do you interpret those indicators on a risk-adjusted basis in a meaningful way?
So I think our polling has reached sort of equilibrium, I guess we could say, and so let's capture that polling data, and we'll bring that forward again with our consensus paper when we do the next step, the revision. And I think that the leading and lagging indicators, I think that academics may actually be a leading indicator because it's the basic research that leads, and the lag is the economic impact, the societal impact, the individual health impacts. Those are definitely lagging, but you can't get to the lagging ones without the leading ones being incentivized, rewarded, and creating motivation, that especially for young people.
Yes, I completely agree.
So we've talked a lot about the pipeline, the end-to-end, going end-to-end, which is research to bench to bedside, and then the return trip. Even more importantly, is what do you learn from where you are at any point in a clinical trial or in a commercialized product that can then feed back and enable the next research questions to be asked, answered, and impact and have even further impact. So, yeah.
Okay, I think the time is over. It was a very fruitful discussion. We move forward. You have time and opportunities to send by email or other options to give your additional comments so that we can prepare the manuscript. Thank you very much. Thank you very much to Glenn and Brennan. It was a fantastic discussion. And the good idea with the poll. Good evening.
Thank you.
Yeah,
good evening.
Thanks also from my side, Brennan and Paul, and we are at the moment receiving even further questions that come in, and that we will then also distribute and include into the discussions that we have for the paper. So Glenn, have a great day. Brennan, have a great day. And I would like to now invite Petra Reinke here to us on stage, and Ronen Rubinov and Uli Dirnagel online, I assume, for the discussion for the wrap-up and the final discussion on the consensus paper draft 2, the role of data reproducibility in translation. And I see Uli Dirnagel and Ronen Rubinov online, and I don't know who would like to start, Ronen, Uli, or Petra, about the sort of the wrap-up of the thoughts that came after the discussion yesterday, after introducing how we came to that point. There were some emails coming forward. You had also some discussions. Any thoughts that you would like to share here? Try to sort of give a little bit of flow to the next consensus paper.
Uli?
Yeah.
Well, I'm not sure how we are going to do this, but I was impressed by at least 2 inputs that we got. And specifically, there was one by Ulf Tölch, which I think is relevant to the discussion, because he pointed to a few, let's say, omissions in our previous discussions and also in the paper. And just to mention 2 of them. One is on replications. Although replication is in the title, it's not featured in the paper. It's not only the problem that it's not defined what it is. I think we didn't bring it up as something that needs to be part of this chain of translation. So somehow I think it has to feature in a recommendation or in a paper like this. The other thing I would bring up has to do, and that I'm just now telling you what Ulf Tölch communicated, and he brought up the issue about the figure, and so in a way the figure has the problem that it does not really specify, and this is maybe also a bit of something we need to discuss. How do we get from A to B? So it's like a wish list in terms of getting to better translation. But for example, on the left side, where it's about this unregulated space, it lists lots of things, but we know they are not there in the unregulated space. We do not get resources for replication. We do not get resources for improving quality with quality management, things like that. So I think what we somehow must also consider and introduce in this narrative is what do we need? What needs to change to get there? So I'll stop here. I think Ulf brought up two very important points, which I think need further consideration in the paper.
Okay, I will highlight another aspect coming from this person, and this was that he states that do we believe or do we mean that the efficacy in animal results is overvalued? And I would say yes, this is one important statement that in particular the efficacy in animal model is overvalued, and we try to figure out many examples underlying these statements. I'm, as a clinician, as a nephrologist and transplant physician, I know for, let's say, 20, 25 years that we are able to tolerize animals, and we we do not achieve any kind of tolerization in human beings. We worked for a long time on sepsis, and even all sepsis models do not fitting to the hypothesis. And now the treatment strategies which are introduced target exactly the opposite, not an acute phase of inflammation, they target chronic inflammatory processes. Also, what I was telling is, and what my strong believing is that the efficacy in animal models is not as trustable as we believe in. The animal models state maybe the mode of action can give us information about molecular pathways, something like this. And they give us information about safety of any kind of possible procedures if we treat the animals, but nothing more. And all the other things has to be done in early preclinical trials, as we discussed it yesterday. And the second point I would highlight from the question is the question of validation. And here I think we have to clarify that validation in the strong unregulated basic research environment is not the same as we use this term in the translational phase. Let's say validation in the translational phase means that we have to define the precision precision of a method, the specificity and the linearity. And if we go to the precision, then it means that we have to have the intra-assay analysis or validation, the intra-assay, the interoperator, and all the things like that, which means it takes more time and more effort as the normal validation will be done or is done in the pure academic validation or repetition of an experimental work.
Yeah, I would agree with that. I think our basic researchers or preclinical researchers are always asking what we on the clinical side at Novartis want to see from a model. And I think I'm always disappointing them that I want to see less than they want to show me. And in fact, I think I want to see that the pathway and target are relevant to human disease.
Yes.
I want to see that the pathway and target are active in the animal model they are studying.
Yes.
I want to see that the intervention they make affects that target and pathway. I think that's it. I mean, it's nice to have pathology. It's nice to have histological confirmation. It's nice to have a disease in the animal. But all of those things will take 2 or 3 more years. And at the end of the day, we're still going to decide whether to go into humans or not based on the original information I just asked for. So to me, let's stop there, figure out the safety, which is another set of experiments in healthy animals, and then let's find out in humans whether it's relevant.
Exactly, and this is a little bit fitting to the discussion we had before with the regulatory bodies, from the FDA and from the PI. And the statement is here the same: start as early as possible, of course, with human analysis, and start, I would say, also in the clinical setting. I'm a little bit struggling with the normal strategy that even in humans we start with so-called healthy problems. For many applications, this is not wise, I would say, and to design the proof of concept in humans has to be associated to the disease or to the hypothesis we have for this kind of new treatment strategy. And this means you cannot prove it in healthy problems mostly.
Just probably one little aspect that while you talked and we had the discussion up front about the direction, one thought that sort of is always coming to me back is all that is true if we know exactly the problem and really understand the clinical necessity and the details. And we're realizing more and more, especially if we go into the clinic, that we sometimes don't know enough and don't really understand the diversity and don't really understand the details. So isn't that all, I mean, it's all true what has been said, but isn't it also depending upon that we really understand the problem good enough? Because if we don't understand the problem good enough, our models can never be good enough because they can only mimic what we know. How much, I guess what I want to say is, how much extra effort do we need to put into characterizing the problem and sort of being a little bit provocative also to Ronan? If you have a big battery in the basement which produces new molecules every other day, and you are then looking, so you produce lots of hammers, and then you have to look for the nail. But should we start with a nail characterization and then look for the hammer? To what degree? It's not always that case, but isn't that to some degree also an issue that we need to consider, because also there is a variability. They all look different, the nails.
So my father, who was a family doctor for 40 years, always told me, medicine keeps you humble. Okay, and the example I can give you of that is, Novartis has 5 blockbuster drugs, over $1 billion a year in sales. None of them is in the original indication for which we took it into humans. Okay, in general medicine.
So that's the concept. That's what you want to say.
So I think the concept is that, yes, you're right. We will, I think it's being adaptive to the data as they come in, and not having that, idée fixe that we only go after the thing we started from, and that it must be a nail because I have a hammer, as you said.
Okay, good.
But that is also fitting to the discussion before, that you have to define clear go and no-go criteria, which is not easy, I know. But also from my experience, you start with a proper hypothesis based on basic research, but the reality is in many cases not fitting to the hypothesis. And I think to prevent further failures and to prevent efforts which do not end up in a proper product development, you have to accept this, and you have to be, let's say, able to acknowledge that sometimes your hypothesis is different from the biology and from the clinical setting. I think this is what we have to learn. And I think also that is important to teach to the young people that this is not a mistake. This is a value to have this strange. Yeah, so also, yeah, okay.
Do you, Uli, want to add anything? Because I think we're coming to a level where we have exchanged ideas and thoughts to that consensus paper. We're coming also to the time limit. I don't want to sort of pressurize to stop, but if you have any additional comments, then this is the moment to make them. Otherwise, I'm very happy. Oh, please.
No, I was just going to say that
Good.
I think we need to continue the discussion offline and finish up the paper.
Absolutely.
And maybe just to say thank you to the people who reviewed the paper and gave us comments, because I think they were very helpful.
Yes, absolutely. So thank you very much, you, for your time, everybody who commented on the papers. And I think we can close then this discussion forum here. And I would like to hint everybody who's in the chat and online, there is a Q&A, there is a last poll that you can answer. Which sessions did you favor? And you can simply click on them and give them an order. At the moment, it's a translational hub story, which I like a lot, but also accelerated translation in times of global crisis, which I find super, especially after the technical challenges that we had in this session. Thank you very much, also Petra. And with that, I think we're wrapping up the current discussion, and we're going to the final comments, and I think I can hand over to Kathleen and ask you for your final remarks and comments from your perspective. Did you like the day?
Thank you, Georg. And first, I would like to thank Georg for all of his wonderful work organizing the symposium. It's been a wonderful 2 days of great discussion. And of course, we need to thank all of the participants for their very active role in contributing to these discussion points. I think also the co-moderators and co-organizers, the moderators, and every speaker and audience participant who really contributed to these wonderful discussions. And from the journal's perspective, I would like to emphasize our appreciation at being able to participate in this very valuable symposium where we can identify those gaps in translation that have, impeded progress from the preclinical to the clinical and even within clinical endeavors towards the goal of translating research findings and medical applications towards the greater patient good. And with that, I need to hand this back over to you, Georg, and we can finish with closing remarks.
Yeah, I have just some very important closing remarks from my side. I'd like to thank Anna Friedrich and Sebastian Schreider from Compact Team, who made that technically possible to have the online meeting, despite the fact that we initially planned a real meeting here in Berlin, having you all here. And we had a great location. We had booked a fantastic location. We had a great team to organize, and then we shifted with that team completely to first to a hybrid format and then to a complete digital format. Thank you very much for that. I need to thank Patrizia Ebel and Stefanie Seltmann from the Berlin Institute of Health for their enormous effort to get us off the ground, get us going, and continue with us throughout the whole preparation phase. I'd like to thank the German Research Foundation, here also today standing for the DFG, Jörg Schulz, but also in the background, Britta Siegmund, and from the Geschäftsstelle Eckhard Picht. Thank you for your support and making that possible, and I wish all luck in the SGKF to continue the discussion about translational hubs. A very big thank goes to Dave Granger, Ola Smith and Kathleen Zakia, that we together with Dieter Volk could get the speakers invited and convince people to participate. And certainly I'm very thankful to all the contributions from you as speakers, giving your talks, being in the podium discussion and joining us for the discussions. And even if it works in most cases without technical challenges, that you stayed with us even when it was getting a little bit challenging getting the voices through. I like to very warm-heartedly also thank Gadi Goldsobel, who was in charge of getting the questions to the panel, Andreas Hüser, and Nicola Brindle, who helped us with the consensus papers writing and sketching as a first draft. And finally, also all the panelists who helped us to get the discussions going, Glenn, Brenna, Petra, Uli, and Ronan. And finally, for funding and making that possible with the budget help from initially Axel Pries, who was very thoughtful thinking about this conference and meeting, and Christopher Baum, who took it on and continued the support with that. I'd like to thank you all and have a great evening, and thank you for listening. Oh, one last point, most important. We have had a voting about the pitch and the best pitch, and I'm sorry, Gadi, I didn't forget it, but I got a good hint. We have a finalist who succeeded and was selected from you. The best video pitch of the challenge translation in Translate 2021 is Sepsis DataNet from Christian Stephan and team of the Kairos GmbH. So we're very happy that you have been voted and selected from the members of the meeting or the participants of the meeting. And we wish all three example pitches. There are much more out there, but we wish all of you good luck and success. And thank you very much for taking part.